Great. Very good. Got super quiet. Good morning. I hope you're all having a good morning. I know it's a busy time. I know when you're at a convention center, you're trying to find your way and get to the right rooms, and it's just not a lot of time. It's a very tight schedule. So good morning, and thank you so much for being here with us. Welcome to the Myasthenia Gravis Foundation of America's Scientific Session, and that's a mouthful. So from here on out, I'm going to say MGFA. I'm Samantha Masterson, and I have the great honor of serving as the president and CEO for MGFA. I've been in this role for almost 5 years now, and certainly I don't need to tell this room how much movement there has been in the space and how exciting it is. It's the most exciting time right now in the myasthenia gravis space. We have a fantastic program put together for all of you for the next 4 hours. We're going to hear about the best science, the most innovative, cutting-edge science and research taking place right now. We're going to learn from each other, and I encourage you, I know that a lot of you are seeing friends and you're making new friends, but I encourage you to really introduce yourself and meet new people while you're here and take advantage of that, because it really is the convening of a global community, which is really exciting. Now, in order to get to the scientific session, it takes a lot of work, and we plan it for the better part of a year. So I do want to thank some individuals who have really given so much time and energy and expertise and guidance to the MGFA, but also in turn to all of you to be here for this fantastic program. So I want to thank our steering committee members. I want to invite them to stand. They're right here in the front. And that's Dr. Castro, Dr. Goyle, and Dr. Booth. Thank you very, very much. I also want to call out that we have some of the MGFA Board of Directors in the room today, and I would also invite them to stand, please, because this is quite a commitment to serve as a board member for an organization. It takes an immense amount of time and guidance and expertise, and just being there and being all in when you're a board member. So Dr. Kevin O'Connor, Dr. Richard Novak, Dr. Kelly Gwathmey, and Dr. Paul Strump, could you please stand? And all serve in a number of capacities for MGFA. Dr. O'Connor, who's going to be our keynote speaker this morning, serves as MGFA's Chief Scientific Advisor, and Dr. Novak serves as MGFA's Chief Medical Advisor. So I know that there's a lot of you who are quite familiar with MGFA in the room, but there's a number of you who are not. And so I'm just going to share with you real quickly so that we can get back on time with the program, that MGFA was established in 1952. We are the only national patient organization solely focused on myasthenia gravis. We have 3 pillars, that is education, advocacy, and research. And we have so many different ways that we are able to bring people together, to convene stakeholders, and to really connect the patient and the caregiver communities, and to make sure that people have the right education and information to be empowered to make the right decisions around their health care. In the last 4 years alone, the organization has grown immensely. We have doubled in size. It's been 2.5 times in size at this point. We've quadrupled what we've been able to fund in research. So it's just been a tremendously exciting last couple of years. So I wanted to make sure that I shared that with you for those of you who are unfamiliar with MGFA. And also, we have 2 major programs each year. I did mention we have lots of ways of bringing people together. We have our National Patient Conference, where we bring the patient and caregiver communities together. And then we have this scientific session, which is our annual scientific symposium. But in the past, every 5 years, MGFA has hosted an international conference. And our last one was in 2022 in Miami. Many of you joined us. The next one is actually next year, because in 2022, we announced that we are going to host it every 3 years based on demand and based on feedback from all of you. So our next international conference is next May in the Netherlands. And I'm just putting this information up there so that you can, and I see a lot of you taking a picture of it. That's good. We want you to join us. So we'll be in the Netherlands, and from here on out, we will host it every 3 years. And you can see the registration information is up there and the abstract submission is up there. We do have our staff, our team member who manages the international conference here. Her name is Laura Chandler. You would have seen her at the doors welcoming you. She is here and can answer any questions that you might have on that event. The other thing that the team did ask me to mention that I want to be sure to be better about talking to you all about when we have programs like this is that we have a program called Partners in Care. It's a network of MG care providers. And there's a vetting process to be officially acknowledged as a partner in care for the MGFA. But if you don't know anything about this program and you might be interested as a clinician who treats patients, I highly recommend that at the back of the room at the MGFA table, there's a team member by the name of Caroline Boline there. Caroline, will you stand up actually? She's raising her hand. She would love to talk to you about becoming a partner in care. We get so many questions about this program. We're always trying to grow it and expand it, and obviously it really just helps the community, helps the patients at the end of the day for us to be able to identify where the care providers are around the country. So if you'd like to learn more, Caroline is here to answer those questions. A couple of housekeeping notes real quickly. The order of the program book, you all should have received one of these. It is going to be out of order in some places. It's not going to be completely accurate at this point because in the last 2 weeks we've had a number of changes. So we do apologize for that, but it was out of our control, and I just want to tell you, I just want to acknowledge that it's not completely in order. Most of it is, but there's a few that will be out of order. And then our poster session. The poster session is in Exhibit Hall B, which is quite a walk from here, but I just want to let you know, Exhibit Hall B, if you go back down the stairs, it's down the hallway where the entrance was, and then take a left, and it's down the only other hallway. It's all the way down there. That's where all of the posters are. There's the formal MGFA poster session from 12 to 12:45, but the posters will actually be left up all week. Just wanted to let you know that. We haven't done that in the past, and the AANEM team offered that, so we took them up on that. So that's really great. I'm glad that your posters are going to be up all week. And then, just letting you know, we have breakfast foods and coffee. It'll be in the room for the first 2 hours of the program, and then if you need the restrooms, go out these doors, and it's right to the left. We're very close by to the restrooms. Okay, and with that said, I'm going to bring Dr. Castro up here, who's going to speak to you this morning and then introduce our keynote speaker. I also want to leave this slide up for a moment. I want to thank all of our very, very generous sponsors who support us in everything that we do. And I know a lot of you are very familiar with all of these companies. We could not do the work that we do if it were not for their generous support, so I want to thank them very much. And they all have, most of them have exhibitor booths around the room, so please go and visit their booths.
Good morning, everybody. This is my last year in the committee. I'm going to miss it. Thank you so much. It has been awesome. And Samantha, I'm going to say something for next year. We need a bigger room because look at all these people standing up. I do mainly pediatric. I do adult and pediatric, but I mainly focus on pediatric myasthenia gravis. So this is an honor, and hopefully I will see you next year. We'll have more to talk about pediatric myasthenia because it's quite important, and we have more research coming. I have the honor to introduce Dr. Kevin O'Connor. He's a very humble man because he said, please make it short, but I have to tell you everything. So he's a professor of neurology and immunology at Yale School of Medicine. He earned his Bachelor of Science degree in chemistry from the University of Massachusetts at Amherst, and his PhD in biochemistry at Tufts Medical School. And he took his postdoctoral training in immunology at Harvard Medical School. His interests are in human translational immunology and neurology, so obviously fascinating, and this is where we are right now in this time of myasthenia, and including prior work on myositis, multiple sclerosis, but his current research is also in immunopathology of myasthenia gravis. As Samantha said, he is part of the Chief Scientific Advisory Board for the MGFA. Thank you so much for being here with us today, and you can come in. Thank you.
Okay, thanks very much, Diana. Thanks everybody for coming. Certainly my pleasure to be here this morning, and I'm delighted to talk to you about some of the work that we've been doing recently on AChR myasthenia. Chip Howard gave a talk this morning at an Amgen-sponsored session, and he called myasthenia a snowflake disease, of course meaning that they're all different. Patients are all different. And what I'm going to talk about today is that the autoantibodies are all different too, and that underlies that heterogeneity. So here are my disclosures. And so let's dive right in to where the disease happens. The mechanisms happen at the neuromuscular junction. And for those of you who may be new, many of you in this room, of course, know this slide very well, that B cells produce autoantibodies that traffic to the neuromuscular junction, and there they impair the signaling of the acetylcholine receptor through multiple mechanisms that we're going to talk about today. So diving right into that heterogeneity, so the autoantibodies that cause AChR myasthenia gravis are heterogeneous. It's been known for a very long time that the mechanisms that they use to mediate the disease are many. And so here on the left, I'm showing you patients with MG, and then through serological studies as well as histological studies, it's been demonstrated that these autoantibodies cause disease by activating complement and then activating tissue damage. They can modulate, which is internalizing the antigen, so they cross-link to acetylcholine receptors and cause them to go inside the cell, so they're not available for signaling. And they can block the acetylcholine from binding to the acetylcholine receptor. So those are three well-known mechanisms of pathology, and I put up on the top right here some of the folks that made these amazing discoveries many, many years ago. The autoantibodies are heterogeneous in that they mediate disease with different mechanisms, but the autoantibodies themselves are heterogeneous as well. And on the right and left, I'm showing you two different parts of an antibody. So on the left side, we're looking at the Fab, which stands for fragment antigen binding. And so that's the part of an antibody that recognizes its target, its antigen, the epitope. And so in myasthenia gravis, on the left side, I'm showing you, there's the acetylcholine receptor on the bottom, and the epitope is the part of a protein that an antibody recognizes, the specific molecular part. There's many epitopes available on AChR, and these antibodies can bind multiple epitopes. So the Fabs, the part that binds that epitope, can be quite different. And then the other end of the antibody is called the FC, it's called fragment crystallizable, and it's the part that mediates effector functions. Most simply, the FC will activate complement, for example. So it's the part that tells the rest of the immune system to respond. And in fact, they're quite different. So on the left side, I'm showing you that the FAB is different. They bind different epitopes. But the FC can be different as well in myasthenia. The FC also defines the isotype of the antibodies or the subclass. So I'm showing you IgM, IgA, and IgG. Those are all defined by the sequence of the FC. And then within the IgG, there's subclasses, IgG 1, 2, 3, and 4. And I'm going to speak about those today. So part one. So how are these heterogeneous autoantibodies represented in the serum among different patients? So are all the patients going to represent these similarly, or are they going to be different? And I think because we've already talked about how this is a snowflake disease, they're going to be different. So work that we've done recently is use a group of in vitro assays, including the clustered cell-based assay, which I'm going to tell you about in a moment, to measure the three mechanisms of AChR-mediated pathology in a group of patients. And this is important, I think, especially in the age where there are a number of new therapeutics available, some of which target very specific antibody isotypes and target very specific effector functions. So here's that cell-based assay. So many of you are familiar with this, but those who aren't, I want to outline it for you because it's a fundamental part of this presentation. You start by presenting the acetylcholine receptor on a cell. So these are cells in the lab. They're called HEK cells, and they're on the left side here where the cursor is. and you're expressing the acetylcholine receptor in its biological form as a 5-subunit membrane channel. And you also express rapsin along with it, and rapsin is a cytoskeletal protein that will cluster that acetylcholine receptor. Clustering the acetylcholine receptor is very important. Angela Vincent and Isabel Leite clearly showed that when you cluster it, the sensitivity of the assay increases a great deal, and patients, many, about 20%, that were formerly recognized as seronegative were actually AChR positive if you use the clustered cell-based assay. Angela Vincent showed that, and this is the gold standard now to use. And so when it's done in the clinic at the Mayo, for example, they will express the acetylcholine receptor, and then they add an antibody to test, and that's here. And then you detect that antibody with what's called a secondary antibody that fluoresces. And then you're using flow cytometry to look for cells that both express the acetylcholine receptor and have antibodies that bound from serum from a patient, and that's your positive. Now, this is done in the clinic with IgG. But what we've done here is we've switched out that IgG for IgM or IgA or IgG 1, 2, 3, and 4 specific secondary antibodies so that we can measure how these are represented in a group of different patients. And so here's where we validated this assay. And I think it's important to point out that this had to be validated. If you buy a secondary antibody off the shelf, off a website from a company that says that it's specific for IgG2, I'll bet you that a lot of them aren't specific for IgG2, and you have to make sure that they really are if you're going to be doing these assays. So what we did here was take an AChR-specific recombinant antibody, and we expressed it with an FC that had IgG1, IgA, or IgM. And then we detected with commercially available antibodies that we validated that are specific only for G, only for A, only for M, and you can see that the M only works when M is presented, the A only works with A. And we did have instances where we bought commercially available antibodies that were supposed to be specific that are not, so this has to be done carefully. And then the same with the IgG 1, 2, 3, and 4, which is much trickier because the sequences of IgG 1, 2, 3, and 4 are actually very similar, so there's very subtle epitope differences, and so there are companies that make very good antibodies that are capable of detecting these 4 different subclasses when a recombinant antibody is expressed using those chains. So you see 1 works with 1, 2, 3, and 4. These are flow cytometry plots of a cell-based assay. Bottom axis is the expression of AChR, and the Y is detecting the antibody. So we have in hand this validated set of assays. And so what we did, we started with a group of patients. We had 46 patients that were very well clinically characterized, and for which we had longitudinally collected samples at very specific time points. These weren't randomly collected. They were all specifically collected at 0, 24, 52, et cetera, weeks, all the same. And so here's the data showing you the representation of IgG, M, and A, and the different subclasses of IgG for this group of patients. It's hard to see there, so I summarized it in this Sankey diagram. And the MAC makes this show up very well. And it's actually good on your screen. It's not good here on the monitor, sorry. But what this diagram is showing you is where all of these antibodies overlap with one another. There's no IgG4, but let me walk you through this. And I'd like to point out, I'm going to show a lot of data today, and this data right here was generated by Fatima Khani. She's a postdoc working with me, and she has a poster right after this where you can see this in much more detail. And at the bottom here, I've also shown a medRxiv preprint that we've published, so this data is available for you to see. But the takeaway is that IgG1 is most frequent, which we would expect. IgM and IgA are present, but they're never present without IgG. You need IgG to get M and A. IgG1 is observed with 2 and 3, and IgG2 and IgG3 also can occur alone. So this represents a pretty clear heterogeneity among these patients in the way that these autoantibodies are represented. So what about the way that they affect pathology? So again, we turn back to the cell-based assay, and this time modify it in a different way. We're expressing acetylcholine on these cells, but now what we've done is we can actually, instead of detecting AChR antibodies in serum, we can detect whether those antibodies, when bound, will activate complement, will block the acetylcholine receptor from receiving acetylcholine by simply occupying the same site, or internalizing the acetylcholine receptor by cross-linking it, causing it to go inside. And so this suite of assays that we and others have developed looks like this. The complement assay has an antibody that recognizes AChR to bind. We add a very carefully tested complement source, and that will activate the classical pathway that deposits C1QB, and we can detect whether or not complement has sat down on these cells with anti-C3b, or you can detect MAC. For blocking, we use a classic bungarotoxin test. So bungarotoxin will bind to the acetylcholine site on the acetylcholine receptor, and you can purchase it where it's fluorescent, and we ask if it will block that bungarotoxin from binding. And the internalization assay is quite straightforward. We add the antibodies, and if they internalize the receptor, the net number of receptors on the surface will be diminished. And so that's how these assays work. And then we applied them to these 46 patients again, and here's another Sankey diagram demonstrating how everything sort of plays out. You can see that they overlap. I mean, the point of the Sankey diagram is to show you what's unique and what overlaps. And you can see that complement activation was most common. So we found in 85% of these AChR patients that we could detect antibody-mediated complement activity. This matches with data that we had produced when we first published the complement assay. So we're quite confident that's a pretty good number. You're a little bit above 2/3. Internalization, or what's commonly called modulation, was present a little more than half. Blocking in our assay was less common, about 30%, and combinations are invariably found. And I just want to point out, I put an asterisk next to bungarotoxin because bungarotoxin is not acetylcholine. It's actually bigger. It's a significantly bigger molecule. So it is used as a proxy for acetylcholine binding, but it's not perfect because of the size of the molecule. So this is what's commonly used, but it's not perfect. So we can estimate that about 30% of these patient serums will block. And combinations are invariably found where you'll see that complement blocking and internalization can also be together, and even blocking can be found by itself. And then quickly, and again, more details are in the paper and in Fatima's poster, what about what happens over time? So over the 96 weeks that these samples were collected, again, all at the same time, you can see, and the point of this is just to show you the distribution being a bit messy. These all change over time. The frequency of the subtypes and the isotypes and the activity of these effector functions will change over time. Importantly, these patients are, of course, treated. There aren't 46 MG patients with palpable disease that are going to be untreated. So this may be an effective treatment. It also may be effective, the immune system varying. So that's an overview of what's happening in serum. And now what I want to do in the second half is dig down much deeper at the molecular level to ask how do the molecular characteristics of AChR autoantibodies influence this heterogeneity? And so the way that we approached this was the production of human monoclonal antibodies, and I'll show you exactly what that is. So what we do here is we start with blood from patients, and we isolate B cells from the blood, we enrich for them, and then we culture the B cells in a specialized media over several weeks. And the bottom line is what will happen is you can differentiate them towards antibody-secreting cells. And within an individual well of a 96-well plate, you will now have media that contains antibody that was expressed by these B cells. And then you can test that media in the cell-based assay that I just showed you, and any wells that are positive for acetylcholine receptor binding are still, the cells are still in the well, and you go back and you clone the cells. I should point out that these cells are exceptionally rare in the circulation, and this process takes a tremendous amount of work. We screen hundreds of thousands of cells just to get one that will bind. And then when we do, we go through the steps of cloning that antibody out by taking the DNA that encodes for the heavy and light chain of the antibody, and we put it into a recombinant system. And at the end, what you have now is an exact replica that you can use in the laboratory that is an exact replica of an autoantibody that was produced by a B cell from a patient. And we validate that in the cell-based assay, of course. And so here's a collection of antibodies. We're up to, and again, 15 sounds like a low number, but to get 15 human monoclonal antibodies from a bunch of different patients took us well over 2 years. It's an incredibly labor-intensive system, but when it does work, you have in your hand the autoantibodies that are from the B cells in these patients that cause this disease, and you can learn a lot from them, so it's worth the journey. So I'm just going to show you 10 of them today. And so what we have here is 10 antibodies. At the top, I'm showing you their somatic mutation profile, what % of the amino acids were changed, and that shows you that these are antigen experienced, they've been through the germinal center, and they've somatically mutated to affinity mature. I'm showing you cell-based assays on the left, but what's more important is their binding profiles. Here they're being tested at different concentrations, high on the left, to the right where they're going lower. And you can see that they have different binding profiles. Some are very strong binders, some are weaker binders, but they all bind very robustly in the cell-based assay. And so now we can start asking some questions about them regarding this heterogeneity that I mentioned. So what is the influence of the FC? I said that the FC was very important for mediating immune effector functions, and one of the most important ones is complement activation. So if you take one of these monoclonals and keep the Fab, which is the red part, the antigen binding part, constant, and keep changing the FC region from IgG1, 2, 3 to IgA to IgM, how does that affect how well this antibody will activate complement? And so here's an example of that. Here's IgG1, 2, 3, and 4 with the same variable region, so it binds the same, but the effector functions are different. And so when we run that in a cell-based assay and look at binding over a range of concentrations, it binds pretty much similarly as you would expect. But then when we put those same 4 antibodies into the complement assay that I showed you, there's a remarkable difference. IgG3 with the same binding Fab is much, much better than all of the other isotypes at activating complement. This is something that's not unknown and wasn't unknown before. IgG3 has a very long FC region that is very good at reaching up to grabbing the first step of complement, which is called C1q. But it depends on the antigen. So it doesn't IgG3 is always not better. It depends on which antigen you're using. So I lost my cursor. Okay, here we're back. Okay, so the IgG3 is better than IgG1, 2, and 3 at complement. But then in the next experiment, again, the same variable region, but now what we've done is asked, what about IgM and IgA? Because I showed you earlier that there's IgM and IgA in these patients, AChR antibodies that use these subclasses. And the difference is here, our winner on the last slide was IgG3, which was really good. And now IgM really is much better than IgG3. Again, not unexpected, IgM is very good at activating complement. But you can see in an assay that measures AChR mediated complement activity that M and 3 are very important and are much more efficient than the other 2 subtypes. So what about the other side? What about the Fab? What if you now ask, how are these effector functions affected by binding? But you would think if an antibody binds to the acetylcholine receptor, it's going to activate complement or do whatever effector function it's supposed to. It doesn't matter where it binds as long as it binds. And so what we did here, because we have a collection of antibodies that bind different epitopes, now we've changed the FAB region. So there's blue, a different antibody, so this is MAB1A, and this is MAB3, and we've kept the constant region that interacts with complement the same. And is there a difference? Yeah, there's a huge difference. And so what you're looking at here is MAC deposition on these AChR expressing cells in the y-axis. On the x-axis, you're looking at all of the different antibodies, negative control here, positive control here. And you can see our MABs. MABs is a short for monoclonal antibody. MAB1, 2, the two brothers of MAB1, A and B. MAB2 are remarkable complement activators, but all of these others are actually very poor, and MAB9 and 10 are very good. These all have the same FC. The FC is not influencing. This is where they bind on the AChR, and they change their ability to activate complement. Here's a really clear example of that. We compared MAB1A and MAB3 together side by side, expressed as IgG1, and the binding, the blue is MAB1A, and the binding is in solid and the complement's in dashed. And they go hand in hand. It's a strong binder, strong complement activator. But then you go to MAB3, it's also a strong binder, but look at its complement. It doesn't do complement very well at all. Same FC, but different binding pattern, and that affects its efficiency at activating this very important effector function in the pathology of AChR MG. What about blocking? Well, blocking turned out to be pretty rare in our set of antibodies. Here's the same antibodies again, and what you're looking at on the y-axis is whether or not we can block bungarotoxin from binding to the acetylcholine site. And only 3 antibodies could do that. Again, MAB1A and B, but then another MAB4 is very good at that. All the others don't bind to the site where acetylcholine binds, so they're not blockers. What about modulation? So modulation here was actually more robust, and again, the 10 antibodies controls. And what we're looking here, we're looking for an antibody that will bind, cross-link the acetylcholine receptor, and then internalize it. And then the way we measure that assay is asking how much acetylcholine receptor is left. We start with a normalized level, and then we ask how much is left after you add the antibody. And we measure that by using bungarotoxin again because it tags the acetylcholine for you. And you can see that these antibodies here, 1A and B, 2 and 3, 3 and 4, I'm sorry, are actually very good at internalization as well as some others. And if you're a real aficionado of these assays, you might say, well, wait a minute. If you're using bungarotoxin to measure the surface antibody, and one of the antibodies is a blocker, you're trying to do 2 assays at the same time and you can't read them out. So Von der Leyen, who is at the Mayo Clinic and is famous for many things, including discovering aquaporin and the disease NMOSD, has a beautiful antibody that we called the Von der Leyen MAB that she shared with us that will bind to a very unusual site on the acetylcholine receptor that human antibodies don't bind to. And we use that for detection, and that confirmed these results, so you get away from the confounding bungarotoxin. But the takeaway after all that technical talk is that these monoclonal antibodies, again, some of them are modulators and some of them are not. And then lastly, what about the epitopes and the subunits? Remember that the acetylcholine receptor is a 5 subunit channel. And there are 2 alphas, a delta and epsilon and beta, in the adult form of this disease. And the antibodies can bind to these different subunits, and they can bind to different epitopes on those subunits. Now, figuring out how an antibody binds to an individual subunit is challenging in the lab. It's been done with many approaches. None of them are perfect. The one that we chose to use, we did in collaboration with Amy Payne, who was at Penn, now at Columbia, and she engineered some T cells to express individual subunits of the acetylcholine receptor. The subunits don't like that because each of their faces need to be touched onto their neighbor. But she engineered it in such a way that she was able to get robust expression, and then we could test them individually. And so I want to summarize where they bind and all of their effector functions in this rudimentary heat map that I made. So here are your 10 antibodies, 10 monoclonals that bind AChR derived from 9 different patients. And red is hot, which means yes, and blue is cold, which means no. And you can see if you go down the list, like MAB1A is really good at binding, very good at complement, very good at internalizing or modulating. It blocks, and it's alpha subunit specific. Same with MAB1B. MAB2 does all of those things except blocking, MAB3 only does blocking well, and you can see the pattern, that these antibodies have these different patterns in terms of their ability to bind well and mediate these effector functions well. And so I don't think it escaped your notice that MAB1A and 1B are really capable of doing a lot of things. And so if you use a Venn diagram to ask whether they can do complement, block, or modulate, they're in the middle. So we call them triple threat. So these autoantibodies derived from a patient can actually mediate all three effector functions at the same time and very well. So you can see it on the diagram here. They're alpha binders, so they cross-link because there's two alphas available for them. And they will internalize and activate complement. And because they're binding on the alpha, where the acetylcholine binding site is partially located, they block as well. So very powerful antibodies. This is a diagram that was first introduced by Tobias Durfus, and then they made monoclonals as well, and then we put ours together with theirs, and you can imagine this type of pattern of where these antibodies are cooperating. So an individual patient is going to have multiple monoclonals that I showed you, not just one, but many. And how do they cooperate? Well, here is a delta binder that can just cross-link two acetylcholine receptors. We're looking at the receptor from the top down, and so it just looks like a bunch of tubes. But then if you have an alpha binder, because there's two alphas, you can actually use, it can chain. And then finally, because these antibodies are going to cooperate, if you have alpha and beta binders, for example, you can actually start to form an array. And this is really important because it's been determined that complement activation favors clustered antibodies that are all facing up and available for the first step of complement, which is a molecule called C1q that kind of looks like a virus. And it has 6 receptors on it, so it's looking for 6 antibodies to bind. And when these antibodies array on acetylcholine like this, they're going to be very efficient at complement activation. So I want to close with one quick story of unpublished data. So here's a patient that we have been following, Richard has been following in his clinic. This is over 96 weeks. And what I'm showing you on the top is antibody titer and then disease score. But what's most important is that as we collected samples from this patient, we did indeed clone one of these monoclonals that I'm showing you here. One of the ones I showed you earlier was cloned. It's a strong binder, and it activates complement, et cetera. What we found by using a technique called B-cell sequencing, where you can find identical B cells with DNA sequencing, we found the same B cell, a relative of that same B cell, at week 72. And so let me show you that on the next slide. The way that you can identify whether two B cells are related is by the sequence of their variable regions, which is highly unique for each individual B cell. One B cell makes one antibody, and it has a fingerprint that is dictated by this complementary determining region where the antibody binds to its antigen. And the green is showing you somatic mutations that were shared between these two clones, but the red shows you new ones. So what happened between time 0 and week 72, this B cell has continued to mature. So it's gone through a germinal center, it's been exposed to antigen and driven by a T helper cell to accumulate more somatic mutations. And when we looked at this antibody in the assays that I just showed you, when we looked at the early one in purple, time 0, and then the red one is at time 72 where the antibody matured, the binding is actually quite similar. But then when you look at the complement by measuring C3d deposition, the week 72 antibody that's more mature was actually considerably better at complement. So the antibody's matured, and its effector function, the way it activates the pathology of this disease, has matured alongside. So this is the big question that I'll sort of leave you with. Is any of this data important clinically? And I think that we could talk about that for a long time, but I'll leave it with this. Chip mentioned, this is a graph borrowed from one of Chip's publications on the use of therapeutic complement inhibitors, and Chip pointed out in our talk this morning that not all of these patients respond to all of these therapeutics. There's invariably a group of patients that don't respond, and we need to find out why. I don't think the molecular immunology that I just showed you answers that question in full, but I think that it partially helps to answer that question. So it's important to remember that therapeutic complement inhibitors do not account for blocking or modulating antibodies, and I hope that I showed you that those are important. FcRn inhibitors don't account for the IgM or IgA isotypes, and I showed you that those are important, and that IgM is actually really good at complement activation. And maybe, hopefully, what we should be doing is profiling the antibody repertoire in these patients before they're experiencing treatment and finding whether or not we can associate these profiles with expected treatment outcomes. So the summary and take-home messages. Circulating serum AChR autoantibody repertoire among patients is very diverse, and it can change over time. IgG 1, 2, and 3 are represented, as well as M and A. And the pathogenic mechanisms that these antibodies mediate are variably represented as well. Some patients have more complement or internalization and blocking. And the reason for that is that the antibodies that are present in individual patients are quite different in terms of their ability to affect this pathology. So the subclass and isotypes can influence that, the epitopes can influence it, and the way that the antibodies cooperate. And so the Fab and FC can impact the pathogenic capacity to a great deal. And I think the easiest takeaway, and Min Pham, who I'll show you on the next slide, who did a lot of the monoclonal work in my lab, pointed out that it's not just the quantity of the antibody. We all know that the titer of AChR antibodies in an individual patient has very little relationship with their clinical presentation. And we propose that it's the quality of the antibody, not the quantity. So with that, I'll close, and I just want to highlight two individuals from my lab. Fatima Khani did all of the work on the serum that you saw at the beginning. She's here, and she has a poster this afternoon. Min Pham is a graduate student that did all of the monoclonal work that I showed you, as well as all the other individuals from my lab, especially Richard, for helping us with all the clinical aspects of this work. Here's my lab currently, and our collaborators from BCMA, Colorado, Penn, Oxford, the Mayo Clinic, and others, and then our funding. And with that, I'll stop, and I thank you for your attention. How are we doing on time?
Yeah, I think we have 7 minutes for questions. Is there anybody?
Kevin, really fascinating work that may help us cubbyhole patients down the road. So I'm going to put IgA aside right now because I just don't know where to put it. Although it makes me think of, IVIG is relatively IgA deficient. All the brands, some of them have basically none. It just makes you wonder about anti-idiotypic effects if there's no IgA there, if IgA is important. But how about IgM? Because the preponderance of complement activity across the board in the patients, and then as you showed, IgM really activates it well. So do we need to even start, first of all, do you think IgM may be important in some of these patients? And if so, do we need to, should you look into subclassifying IgM with heavy chain differences and the involvement of heavy chain in activating complement, I think? Yeah.
Yeah, it's a really good question, Gil. I think that the data that we have now suggests that IgM is present in a rare group of patients. And then in those patients, you're not going to find it without IgG. So then the question becomes, is the IgG at a higher concentration than M? So I don't have a clear answer for you. I think IgM is certainly important. I think that as we continue to do research on these subclasses of antibody and isotypes, that we're going to identify a small but unique set of patients for which these particular isotypes are of very, very high importance. Small group, but they're going to be critical.
So that was really, really great work. I had a quick question. So I hope I get this right, but basically you go from where it's a B cell population, patients have myasthenia gravis, and you generate these monoclonal antibodies, right? And these are individual monoclonal antibodies, obviously. And have you been able to go back and see what proportion of the total antibody in the patient these monoclonal antibodies represent? In other words, are they rare? Are they common? Are they
Right.
frequent? Because you could have like a very low frequency of a super pathogenic antibody versus, and we know there's a diversity in the number of antibodies.
Right. It's a very, very good question. Thank you for asking it. It's a technical hurdle that we are addressing that we've not completed yet. And essentially, when we isolate one of these monoclonal antibodies from a patient and we're doing these in vitro tests, do we know that that one monoclonal represents the entire circulating antibody repertoire in that individual patient? And the answer is no. We don't know that yet. We would have to make all of the monoclonals from that patient, and because it's technically challenging, we're not there yet. We recognize that, so we're just studying these individual molecules by themselves, and I think that their presence in the wider circulating repertoire is important to consider. Thanks for asking that.
Thank you for the elegant presentation. So in the clinic, sometimes we see patients with thymoma and positive binding antibodies, but no clinical symptoms of myasthenia gravis. So does the presence of thymoma influence the diversity of the acetylcholine receptor antibodies?
It's another great question. Thanks for asking it. It's something our lab has just started working on. We have not started to isolate monoclonals from patients with thymomas until just recently. And so I think that one of the questions we're asking is, are the autoantibodies in thymoma patients different in terms of their properties and characteristics than those that are present in the more classic autoimmune derived disease? So I'm sorry that I don't have a good answer, but it's something we're looking at. But it's a great question, and they do look, these patients present differently. We need to figure it out.
Great talk. Two questions. One is, there are commercial assays looking at modulation, blocking, binding, and when you look at this individual patient antibody profile, do you see any correlation between the monoclonal you isolate from that patient versus the result from the commercial lab?
It's a good question. We didn't test it. We don't use a commercial assay in our lab, and we haven't endeavored to compare the clustered cell-based assay side by side with the radioimmunoassay, so we haven't looked. We decided to use the clustered cell-based assay because we think it is the best representation of the biology.
The other is, there is a main immunogenic region in the alpha subunits. And with your data, does that concept still hold true? We're just curious.
It's a great question. So we're a lot of MG people here, so I'm gonna say something. I don't think the MER is important as everyone thinks it might have been. I think there's a lot of historical literature that says the MER is the binding site of the acetylcholine receptor antibodies, and it's been accepted as such. I'll tell you that our data, I only showed you 10 antibodies, we have more. We don't find that the MER is the one that is most often targeted, and we find plenty of monoclonals that don't bind the MER that are really good at pathology. So I'm not married to the MER, and our data will hopefully continue to dissuade the dogma that's attached to it. And as a scientist, I reserve the right to be completely wrong. So we'll see.
One last question.
I'm struck by the fact that the antigen specificity can affect complement activation, even at the perhaps relatively subtle level of the affinity maturation changes that occur with time. Do you have a potential explanation or mechanism for why it would affect complement activation?
I think that so that's a really good question. I think obviously the immune system is not testing complement when it matures an antibody. It's testing binding. I think that in that assay, what we're seeing is an antibody that probably binds a little bit better. I know that the cell-based assay that I showed you on the left side of that graph showed that those two monoclonals bound similarly. We haven't done affinity measurements yet, and I bet if we do, they're probably going to be subtly different. But I'll leave you with this. it's not only how strongly they bind, but the architecture of the antibody when it binds. And subtle changes in amino acids in the CDR3s can actually shift the FC in the way that it's facing up from the cell. And that has a very important influence on the way that C1q binds. And so it's possible, and again, without structural work, we wouldn't know, that when this antibody is maturing, it's changing the way that it binds to favor complement activation.
Please, another applause. This was an amazing, amazing lecture.
Thank you.
Now we're going to talk about biomarkers and basic science. We have our first talk about plasma biomarker-based endotyping reveals distinct inflammatory patterns in myasthenia gravis, and is Dr. Zhang. Please come in. Do you mind introducing the next person?
Sure, of course.
Okay.
Okay.
Just gonna leave it right there.
Good morning, everyone. Find my slide. So my name is Hua Zhong from Huashan Hospital, China, and it's my honor to be here and to introduce our previous work on plasma endotyping in different myasthenia gravis subtypes. So this study originates from a simple question. Is hypercytokinemia a common feature of MG? And because my lab mate, Dr. Huan Xiao, several years, she followed some myasthenic gravis patients, and she found that the pro-inflammation cytokine levels actually fluctuate quite cooperate with the disease severity. And at the most acute stage of myasthenia gravis, this level usually very high. And our lab last year, we did some single-cell sequence analysis in myasthenic patients, and we found that apart from cytokines, chemokines may also play important roles in this disease pathogenic. And we found that many other studies also indicate that both cytokine and complement, they contribute to disease, and they fluctuate with disease severity. So we carry out this plasma endotype investigation in different MG patients. The patient source were from our patient registry, and we selected this about 200 myasthenia patients of different antibody types, and basically we send them plasma for Luminix protein assay of 22 cytokines and chemokines and complements. So we do some dimensional reduction analysis and pattern recognition to try to find the interconnection between them. So it turned out that in myasthenia patients, these cytokines, their levels are very high compared to healthy controls. And we found that actually TNF-alpha and C9 correlate mostly with MG severity. So we look into different myasthenia antibody types, and we found that AChR MG patients, they are featured by highest level of interleukin-6 and C9. And on the contrary, MuSK patients, they featured by lowest level of BAFF, B-cell activating factor, and seronegative MG is featured by highest level of interferon alpha and CCL3, a chemokine ligand. So we try to mapping these cytokines to different cell type responses according to literature knowledge. And we found that in AChR MG patients, almost all, all cell type responses are very high. And in mask patients, they exhibited a milder endotype. And in seronegative patients, most cell type responses are very high, except complement response. But it has a very different emphasis compared to AChR patients. So we next, because we know that plasma have 22 features, so we send them for clustering, try to find different groups. And we have generated 3 clusters. The cluster A is mostly from post-immunosuppressive patients. They are lack of complement response. And cluster B is mostly from naive patients, and they show high complement response. And cluster C, and they mostly from healthy controls. And these cell responses are very low in all respects. Because we found that actually treatment is related to this plasma endotype, so we next did some retrospectively, did some MG subgroup analysis on these 68 naive AChR MG patients. And we used them baseline plasma endotype to predict their treatment response of steroid after 1 year. And we found that high levels of baseline interferon alpha, interleukin 23, interleukin 2, CXCL2, 4, and 5 indicated unsatisfactory steroid response. So the red group is SNR, which means steroid non-responder, and the yellow group is SR, which means steroid responder. So we postulate that patients with elevated level of interferon and pro-inflammatory cytokines may be at high risk of disease progression, making them more likely to require escalated therapy beyond the steroid. So take-home message, we found that actually MG patients, they have a cytokinemia feature, and including also different chemokines and complements. And we found that AChR is characterized by highest interleukin 6 and C9, and mask is by lowest level of BAF, and seronegative characterized by highest interferon alpha and CCL3. And some plasma biomarkers is closely associated with the drug response of these patients. And of course, this study has many limitations. First is that the number of patients in mask and SNMG groups is limited. And second is that the conclusion of cell type responses were mainly based on literature knowledge. So there's no flow cytometry experiment conducted. And this data were mainly from Han Chinese patients, so the results may not be fully applicable to other ethnicities. So finally, we want to thank all patients and their families, and we also thank our thoracic surgeon and collaborator and lab technician Mingji. And this study is also supported by some grants from China foundations. Thank you. So please let me introduce the next speaker, Dr. Anna Ponga. Please welcome.
So, thank you very much. Good morning to all, and thank you for the possibility to present two of our recent studies here. So, two different abstracts. I will go through them one at a time. It's so nice to see a lot of happening in MG, and we'll continue that track now on a little bit on the immunology/biomarker trail for MG. Disclosures. So the aim of the first study was to see whether we could define a serum protein profile which would separate AChR positive MG patients from healthy controls to further validate these as MG biomarkers. And that is due to the lack of objective biomarkers that we have today. The secondary objectives was to assess whether these inflammatory proteins were altered in the different subgroups of early onset versus late onset MG, and also in patients with immunosuppression versus no immunosuppression. So the cohorts we used, in short, you see in the table here. So we had 98 AChR positive MG patients from Sweden and Latvia through the collaboration with Riga University Hospital. And we also had matched healthy controls, 77 healthy controls. The patients had a duration median of 6 years, and MG-ADL was ranging from 0 to 16. We had most of the patients, which can be a limitation in some studies, on immunosuppression. So about 70% on immunosuppression. And we tried to have about half of the group in LOMG group, 47%, and in EOMG group. And what we use is a technology which is very much used in Uppsala through Olink platform, where we use this proximity extension assay with a very, very sensitive assay, antibodies targeting different parts of the proteins to really determining very, very low concentrations in serum samples. So the results, so you see here in a very, very, I'm sorry about the small resolution here. So we have tried to map down the most significant proteins. So since we assayed a lot of proteins, 92 proteins, we had to use a very, very low p-value, so less than 0.0006. Despite that, we did find 16 proteins significantly higher, and you see the ones here. So blue is MG patients and green is healthy controls. And we were quite amazed that they were actually totally separated between the MG patients and healthy controls. 7 were significantly lower. And among those that had the highest difference, of fold difference, we found one of the chemokines, CCL28, and then we found the fibroblast growth factor, FGF23, FGF5, TGF-alpha, and in the TNF superfamily, TNF-SF14, and UPA. And we worked closely together with statisticians because this is a work trying to map down how we can validate these now with the difference between MG patients and healthy controls. They use something called the Baruta algorithm to really plot the proteins as the fold of importance and the magnitude of importance for all of these 92 proteins. So in this long row here, you see, and this is an algorithm where you feed the data to this program, and then it comes up with trying to repeat the expression over and over again to see, does this hold as a marker for MG versus healthy controls? And we came up with these proteins in green, and again, the ones being mostly associated was CCL28 and TNF-SF14 and so on. This is all published. You can read for those of you interested very recently. And also we wanted to see if we use a number of these proteins, can we then separate MG from healthy controls? Because that would be one of the points in trying to have some kind of signature protein markers for MG versus healthy controls. This volcano plot here down at the left, but also in the top here to the right, you see if we choose the top 20 of these protein biomarkers, we can have almost a clear separation between healthy controls and MG patients. We also wanted to look into the subgroup profiles of EOMG versus LOMG here in A, and we found that 2 proteins, we would have anticipated more actually, but you know, sometimes you're biased by your previous thoughts. So we did find TGF-beta1 and OPG significantly separating themselves between EOMG and LOMG. So maybe sometimes the subgroups we choose are not the actual ones which we always should choose. Regarding the patients with immunosuppression versus those without immunosuppression, we also found some that were clearly separated. Oh, those were very sensitive here. CXCL10, CCL11, IL-17C, TGF-alpha, and TNSF14. And we also found one that separated patients with a mild disease based as MG-ADL 0 to 4 versus MG-ADL over 5, and that was UPA. So summarizing this in the figure here to the left, so we tried to map all these proteins into families of action and also how they go together. What we also did, but I didn't have time to go into here, we mapped them down with previously found microRNA, especially microRNA 35P, 155P, and 21,5P. And we did see that there was a, I don't know if you see my pointer here, no, maybe not. They also had a large correlation between these microRNAs and the proteins. So in summary, we managed to define a profile of inflammatory serum protein biomarkers separating AChR MG patients from healthy controls. And the data indicate the importance of chemokines, interleukins, TNF superfamily, and other pathways in AChR positive MG. And we really are very grateful for the support from MGFA now with a high price grant to be able to do the validation study for this, which is ongoing. Great, on time, so study number 2. Study number 2 is a little bit in parallel with the first one, but this is now to also go into the complement system and visualize the membrane attack complex in MG patients. So aims were to analyze complement activation products C3A and C5b9 and 6 native complement products in plasma and sera in parallel from MG patients and matched healthy controls. And also we performed in vitro human disease modeling by having human muscle cells and also attaching sera from MG patients. So this is summarizing the method. So we had serum and plasma, as I said. So the whole point is complement factors are more precisely measured in plasma because you stop the process of activation of complement, which is not stopped in sera. Whereas we use actually sera for the functional assay where we have human muscle cells and apply the sera to see the activation of the complement system and the attack of the membrane attack complex. So this was our cohort. For the serum samples, we had healthy match controls, and for the plasma healthy controls, we had a larger group because this was based on a lab healthy control cohort in Uppsala. So it was a smaller cohort, 23 MG patients, and this is the characteristics. So most of them were AChR positive. We did also have 2 who were double seronegative, and they had the MGC's median 6.6, and a disease duration, which in some cases were quite long. So the results, even if we had the limitation of having quite few samples for this analysis study, we did find very significant data, especially regarding C3A and C5b9, as you see in A and B here. Not in serum as expected, but in plasma. So we do believe these factors should be checked in plasma, which can also explain some different results in the literature. So C3A was very much higher in plasma in MG patients compared to healthy control, and so was soluble C5b9, that is the S C5b9. We also found higher levels of C5 in MG patients, although not as high levels as the other two. And also another factor separating was factor I. And factor I was higher in MG patients both in plasma and in sera. And properdine is a factor which was lower in plasma of MG patients compared to healthy controls. So then we wanted to see, with the limitation that we didn't have any other patient groups here to really say something about the true sensitivity and specificity, but compared to healthy controls, we found, especially for C3a in plasma, sensitivity for 95.7% and a specificity of 100%. So with the caveat, as I said, we did not check any other disease controls. And the soluble C5b29, which is the membrane attack complex, but soluble form, had a specificity which was 96.6%, but a lower sensitivity. We also did find correlation with the MGC, so the MG composite score for some of these factors, especially for plasma factor B in A, plasma factor I, and plasma factor H. Then what I thought was the most fascinating part was really to visualize the membrane attack complex, MAC. So these are the primary human muscle cells cultured in our lab. And then we wanted to, of course, activate the AChR clustering, so we have added agrin as well according to the standard protocol. So in healthy controls, we see red is here acetylcholine receptors, and in green, we should see, what we don't see in the healthy controls, the MAC, so antibody against C5b29, and desmin is blue, is coloring the muscle fibers. So we had two different timelines, half an hour and 2 hours, just to be sure what we captured the process. It's a very fast process. So in the non-treatment MG patients, these are examples of images. So after half an hour, you see already the deposition of MAC, which is also more prominent after 2 hours, a little bit low resolution. And also we saw this in the prednisone group of patients and also in the azathioprine. So pointing here, I will show you larger in the next one. So we also looked into the number of AChRs in these different patients, and we saw that it was really a difference to the healthy controls in the number of AChRs available at the muscle cells and lower in the non-treatment and also in the azathioprine groups. We also found a difference in the deposition of MAC with the lower level in the azathioprine group than the non-treatment group, but we didn't see that in the prednisone group. So this is again zooming in. So healthy control, as I said, you only see AChRs and blue muscle cells. And in the non-treatment MG, you see these almost encapsulated AChRs by the MAC here in green. It's very beautiful, I have to say. It's almost like a moon landing. So here in green, you see the MAC surrounding the AChR, almost engulfing them. So the next step are now we are trying to understand what are they doing, what is happening with the AChRs. Are they exocytosed or what is happening? So this we managed to really zoom in and look at this. It really encircles the AChRs. So summary. Plasma C3a and soluble C5b29 are promising biomarkers for future validation in MG to assess complement activation. And visualization of MAC deposits proves that the complement cascade is activated on muscle cells after applying the MG patient sera to the muscle cells. And suggested also the role of MAC in the observed AChR loss. And we see this, as I said, not only in patients who are treatment naive, but also in patients with azathioprine treatment and prednisone treatment. So with that, I also like to thank our lab in Uppsala, especially Yufang Huang, Amul Bandage, who has been very much involved in the studies, and our collaborating partners in Riga, all the MG patients involved, international collaborators, and thank you all for the attention and for the MGFA for further supporting our important biomarker works. Oh, you do? Yes.
Thank you so much. The next talk is imbalanced pro-inflammatory immune response in seropositive and seronegative MG patients, and it's Dr. Lee.
Hi, good morning, everyone. Today, I'll be discussing a pilot study by using single-cell RNA sequencing to explore immune cell dysregulation in Myasthenia gravis. Primarily focusing on two subtypes, AChR MG and seronegative MG. So the definition of seronegative MG is double negative for AChR and MuSK. So MG is an autoimmune disease that affects the neuromuscular junction. The immune system plays a central role, but the traditional methods like flow cytometry fails to capture all the immune cell subsets simultaneously. So that's why we use single-cell RNA sequencing to get a clearer, more unbiased view of both adaptive and innate immune cells in MG patients, and then compare them with health controls. For this pilot study, we include 2 AChR MG patients and 2 seronegative MG patients. The PBMC cells were isolated, and then dead cells removed, followed by library preparation and sequencing. And in the visualization, each dots on the map represent an individual cell, with the cell clustering according to the similar mRNA expression patterns. And to minimize the confounding factors, so we selected patients with age, gender matched, generalized MGFA classification 2B, and without immunosuppressant treatment. For the seronegative MG patients were validated double negative for AChR and MuSK by using radioimmunoassay and live cell-based assays. So due to the cost constraints, approximately $6K per sample, we only include 4 MG samples in this pilot study. In addition, we included 24 health controls data from the public dataset. So when merging MG patient data with health controls, we identified and annotated 30 distinct cell clusters in PBMCs, giving us very high resolution view of immune diversity. So we can detect monocytes, NK cell, CD4, CD8, and B cell groups from the PBMCs. So when focusing on the innate immune cells, the monocytes can be categorized as classical, intermediate, and non-classical based on the CD14 and the CD16 expression. And we found that compared to the health control, monocytes from the MG patients can exhibit different CD14 and CD16 expression. So it indicating that it's shifting towards the intermediate state, which are associated with the inflammation status. So when we extracting the monocytes from the whole PBMC cells, and then we reclustering the monocytes. So the subgroup analysis reveals that increased pro-inflammatory of monocytes and decrease of the inhibitory monocytes in both AChR, MG, and seronegative MG patients. But we didn't find a significant difference between the 2 MG groups. Very similarly, within the NK cell population, we found that MG patients had a significant decrease in inhibitory NK cells. So this pointing towards a potential loss of immune regulation in innate immune cells. When shifting to the adaptive immune cells, we found MG patients have increased pro-inflammatory CD4 T cells and decreased of regulatory CD4 T cells. This further indicating an imbalance between pro-inflammatory and regulatory CD4 T cells in MG patients. So in the CD8 T cells, the MG patients also showed decreased regulatory of CD8 T cells. Specifically, in seronegative MG patients, there is also a decrease in functional CD8 T cell compared with the AChR MG patients. The functional CD8 T cells are very crucial for the immune response. And finally, in the B cell population, we saw an increase in the activated B cells in MG patients. So especially in the seronegative MG patients, these activated B cells have markers indicating higher level of inflammation and migration potentials. So in conclusion, our pilot study reveals a distinct shift in immune cell profiles between MG patients and health controls, with increased pro-inflammatory and activated immune cell subsets. These changes are particularly evident in the median monocytes, inhibitory NK cells, CD4 and CD8 T cell, and B cells as also. And importantly, in the seronegative MG patients, shows a very unique reduction in the function of CD8 T cell and more activated B cells. So these findings provide a new insight into the immune mechanism underlying of the MG and may guide future therapeutic strategies. Last but not least, I would like to thank our team, collaborators, and funding resources for their support in this research. And thank you for your attention. So next, please welcome Dr. Sikowski.
Okay, good morning, everyone. I'd first like to thank the organizers for the opportunity to share my work with you today, which will focus on alterations in peripheral B and T cell subsets in MuSK-MG after rituximab treatment. B cell depleting therapy, rituximab, that targets CD20 has been widely used to treat antibody-mediated autoimmune disease. In MuSK, rituximab has been shown to be beneficial, showing a better and more sustained response compared to acetylcholine receptor positive MG. And efficacy is thought to result mainly from its impact on short-lived plasma cells and the subsequent reduction in autoantibody levels. While many subjects show clinical improvement, disease reoccurrence is common, and this can occur this can result from primary failure due to the failure to deplete long-lived plasma cells that lack CD20 expression, as well as insufficient depletion of B cells in tissue, such as the spleen. And more recent studies have also described the presence of resistant memory B cells after treatment. These resistant cells, as well as newly emerging naive B cells, contribute to relapse after this primary response. Activation of these cells through follicular and extrafollicular pathways are postulated to drive plasma cell production, contributing to disease reoccurrence. However, the exact mechanisms contributing to long-term remission and relapse in MG are not completely understood. So to better understand the factors contributing to treatment response in MuSK, our objective was to evaluate the immunological effects of rituximab. Because B cells have additional functions such as antigen presentation and cytokine secretion that could significantly impact the T cell response, we expected that characterizing changes in the T cell compartment after treatment would provide additional insight into mechanisms contributing to clinical efficacy. That leads us to our long-term objective, which is to better understand the cellular mechanisms of anti-CD20 treatment in MuSK, contributing to long-term remission and relapse. For this study, PBMCs from 6 subjects were analyzed using multi-parameter spectral flow cytometry. PBMCs before treatment were compared to those PBMCs collected 4 to 15 months after treatment. 2 patients were in remission, and those are color-coded in orange throughout the presentation, whereas 4 were symptomatic after treatment. By comparing these groups, we wanted to assess if there were any trends in the data in terms of treatment response. As expected, we observed a significant reduction in total B cells after treatment. In terms of B cell subsets, we didn't see an overall change in naive B cells. However, if you notice, the two subjects in remission did not show changes in levels of naive B cells, and the frequency of naive B cells were higher than those that were symptomatic after treatment. Treatment also significantly reduced switched and unswitched memory cells, whereas double negative memory B cells were significantly expanded after treatment. Notably, the effect in the double negative compartment was not as prominent in the two subjects that were in remission. The presence of double negative cells after anti-CD treatment has previously been reported and associated with relapsing conditions such as RA and MS. Double negative cells can be divided into subsets using markers CXCR5 and CD11C, and we were particularly interested in the double negative 2 and the double negative 3 subsets because the increase in these cells are associated with autoimmune disease. Double negative 1 cells, which are most commonly found in healthy individuals, were significantly reduced after treatment. We didn't see an effect in the double negative 2 compartment. However, we did see a significant increase in double negative 3 B cells. And this subset is of particular interest to us because recent work done in our lab has shown that MuSK subjects that aren't on any immunosuppressive therapies have significantly higher levels of double negative 3 B cells in circulation compared to acetylcholine receptor positive MG, as well as non-autoimmune controls. The expansion of double negative 3 B cells is associated with the extrafollicular response, which is one pathway that can lead to rapid differentiation into short-lived plasma cells. Given that these short-lived plasma cells are thought to be the primary producers of MuSK, we further evaluated if the B cells after treatment show evidence of extrafollicular responses. And what I'm showing you here is an expression pattern of several markers that are consistent with the extrafollicular response. And that includes increased CD11C, CD95 expression, and reduced expression of CXCR5, CD21, and CD19. We are currently working on understanding the relationship between these extrafollicular responses and short-lived plasma cells in MuSK and their role in treatment response. We next evaluated the impact of rituximab on T cell populations to gain insight into B cell influence on T cell responses. Analysis of total CD4 positive T cells shows that they are significantly increased after treatment. However, no changes in Tregs or T follicular helper cells were observed. In the naive and memory compartments, we saw opposite effects. We saw significant increases in naive CD4 T cells and significant reduction in terminally differentiated effector memory cells. Similarly, in the CD8 compartment, treatment increased naive CD8 positive T cells and reduced effector memory T cells. These findings suggest that B cells support both CD4 and CD8 T cell responses in MuSK and may contribute to treatment response. To summarize these findings, we found significant alterations in B cell and T cell memory compartments, specifically in the downregulation of unswitched and switched memory B cells, and also effector memory cells in both the CD4 and CD8 compartments. We saw a significant upregulation of autoimmune-associated double negative memory B cells, specifically the double negative 3 cells, and extrafollicular responses, as well as an increase in naive CD4 and CD8 T cells. This provides evidence for B cell and T cell collaboration that may be important in treatment response to rituximab. We're currently working on analyzing more samples to correlate these profiles with clinical response, as well as longitudinal analysis to follow these populations over time and define mechanisms driving disease reoccurrence. And with that, I'd like to acknowledge my mentors, Linda Kushner and Henry Kaminsky, as well as members in our laboratory and our funding sources. Thank you.
We have only time for maybe 2 questions for this session. Everybody wants the break now. Yeah, no questions. Okay, so let's do the break. Thank you.
Hi, how are you? Good, how are you? I was interested in talking to you about the program. Yes, one of the child neurologists.
So we would love to have you be part of the program. So what I can do is I can get your email address
Sure.
and then send you the application. So it's an application, but it's a formality to make sure that we have all the information. It all goes demographic details, and we'll ask how many patients you see, those sorts of things.
Cool.
And then we might have speaking opportunities that we can have. We'll send you in the future a package of materials that you can distribute to your patients, especially pediatric patients. We have specific materials. So we have a new care team with MG support group. So one question. Details, and then really what you'll get every month is a newsletter that's just like, I try to keep it the most critical updates so that you're not having to sort through all of our emails. You can just get like one email a month that shows you everything you need to know.
Awesome,
So,
yeah. Because I run the pediatric myasthenia gravis consortium.
okay.
So we've been working with MGFA for a long time.
Yeah.
And I work with Dr. Castro.
Fantastic. That would be great. Let me get your email address. And I will send you an email tomorrow, actually, when I get back to my desk.
Okay.
And that way you can have the information and get you anything you need. Hi, how are you, Shree? Good to see you. Yes, that's right. Shree's on the Partners in Acute Care Council.
Hi.
So.
Nice to meet you, John.
Yeah.
Hope you can make my hand.
I will figure it out for sure. So
Awesome.
thank you so much. I will be emailing you
All
soon.
right, nice to meet you.
Nice to meet you too. We got one. Oh, it definitely. Yes, it'll be great. So, hopefully, yeah, I need to talk with him. Send him my way to talk. But it's so good to see you in person. Yes. Sorry? I'm good.
Perfecto.
Wow.
I know. Yeah, that's right. Exactly. Perfect. Perfect. Okay.
From Boston.
Boston.
So I'm just trying to learn a little bit more about MG. It's kind of like one of the therapeutic areas that we might be interested in. So,
Are you a clinician?
On the biotech side.
Okay.
So we're just kind of beginning all of this, and I was like, this is going to be a great opportunity to kind of start to learn a little bit more. So really wanted to thank you guys again for putting this session together.
Well, I'm glad you're enjoying it. It's so nice to meet you.
Yeah, nice to meet you.
Yeah, this is my first time at Scientific Session too, so I feel like I'm learning a lot.
That's awesome.
Yeah.
Did they give you a shout out?
Yes. So I oversee our representation program. So I like all our neurologists that partner with us. So yeah, I was excited. It's not, of course, it's a smaller group of our team that are here.
Yeah, you guys have grown so much since each
Yeah, I think we have 13 or 14 on our team. So.
That's awesome.
Yeah, so good. Glad to be here and listen in. Some of it's over my head, but I've enjoyed listening.
Yeah, I mean, I think that the clinician part of it is so important because, you know, with such a diverse disease, like you want to be able to have all the information rolling in.
Absolutely, and it helps us to reach patients in various country, and so it's really important, and we're growing. In the past, it's functioned more as like a network, but now it's really kind of a community. It's been that way with a lot of countries for a while, but it's becoming more and more like that. Yeah, it's really exciting.
Thank you.
Yeah, so great to meet you, and yes, definitely.
All right.
I'm not a physician. I'm in because of research into PMG. Basically, a lot of it's serious young disease. And I guess this is actually quite interesting. Kind of addressing one of the things that we're quite interested in. Therapy. So, healthy, just keep track of this. Like this, is that being
It all works.
Yeah. Great.
Slide advance here on the left.
I mean, are we able to see if slides are on there, or?
Yeah. I mean, if you double click, although I think it's probably going to show up for everyone.
I don't want for everybody. That's what I was trying to.
How are you? Nice
The good news is they're there.
Did you get your materials?
That's all I
Okay,
need.
good. So we're working on materials for next year, which will be a little bit more comprehensive, I guess, of all the programs and things that we offer. So we've got it in our budget for next year, so we'll probably send it out either December or January, but it'll be a full package. But hopefully that will hold you over until, and then if you need something else specific, please let me know, and I'm happy to. Oh wow, okay.
Yeah, that'd be great, yeah.
Okay, I'm glad that you got them, because sometimes it does take our printer a little bit of time to get them out the door, so. Yeah. Oh, good. Okay, great. Well, if you need anything else in the meantime, let me know. But I think next year we'll have some folders that will have all the materials inside. So all you have to do is you have to go to your patient folder, and it'll have have everything they need in there. And then we'll have a poster that you can put in your practice as well. So hopefully that'll be helpful too. Yeah. Not quite there yet, but it's being worked on. It's more on the research side versus the clinical investments. I know that's about all I know. It's kind of in discussion with the board and but actually Laura knows more about it, so I can ask her to share a little bit more. But probably 6 months out.
Yeah, this is really cool.
Oh wow, okay. Okay, yeah. Right. Right, yeah. There's nothing else as of right now, but hopefully. So, yeah, and Laura, if you have a chance, she can share a little bit more, but I think it's very much in the not brainstorming phase, but I don't think any decisions have been officially made. So, yeah. But we'll definitely keep you posted, and as soon as we have something to share, I'll send it out to all of our workers in here. So, yeah, nice to see you. Yeah. Please take as much as you'd like. Yeah.
Yeah, I think it's very like
Please, yes. Citizenship thing would be quite
that's gonna be great. It's gonna be a great like
don't remember his name. I'm gonna go snag him up and not you want one?
I'll just welcome everyone back to their chairs so we can start promptly. All right, everyone, we'll have you come and take your seats. I'll be introducing the next section, which is therapeutics. Very exciting. All right, it's my pleasure to kick off the next section, which will be focusing on therapeutics, and I'll invite Dr. Richard Novak to begin the session.
Great, thanks so much. I'd like to thank the meeting organizers for inviting us to present the top-line data for MINT. We're really coming full circle. I presented the study design, the rationale for the myasthenia gravis and immunopathology trial actually at this meeting several years ago, so many of you might remember. So we are coming full circle as this is the first top-line data presentation for MINT. Couldn't do this trial. It's really a global effort. I say that, you know, it takes a village. Many of us say that, but this truly is on the shoulders of the MG community, the MG investigators throughout the world. So it's my privilege on behalf of all investigators and the entire team to present our results today. So these are relevant disclosures. I just wanted to note that the study was funded by Amgen. And just a brief introduction is that myasthenia gravis, and you've heard this already, is mediated by pathogenic autoantibody producing CD19 positive B cells that target critical proteins at the neuromuscular junction, leading to essentially muscle weakness and fatigue, or fatigable muscle weakness. Inebilizumab is a humanized monoclonal antibody that causes targeted and sustained depletion of CD19 positive B cells, including specifically plasmablasts and plasma cells, which are integral to the pathogenesis of generalized myasthenia gravis. So as you've also heard earlier this morning and from Dr. Sikorsky, is that the autoantibody producing B cells are really plasmablasts and plasma cells. So if you can see down here at the bottom, we talk about the antigen expressed on B cells, and clearly CD20 expression on B cells becomes lost as B cells mature. So the general idea was, when we looked at our prior data, especially with CD20-directed depletion therapy, is that there's been mixed results in the literature about whether or not patients truly benefit, which groups benefit more, et cetera. So inebilizumab had a lot of rationale in terms of understanding and investigating it further in the context of a prospective placebo-controlled clinical trial. So that's what MINT is. It's a phase 3 clinical trial that was conducted to evaluate the efficacy and safety in antibody-positive generalized myasthenia gravis. So this is our study design. So in terms of screening, there were just under 500 individuals screened for the study. And there was a one-to-one randomization to study drug inebilizumab versus placebo. There were technically 2 cohorts of patients enrolled. The acetylcholine receptor autoantibody cohort that had a blinded period of up to 52 weeks, and the MuSK antibody cohort that had a placebo-controlled blinded period for up to 26 weeks with an optional open label extension period that lasted up to 3 years, it really depending on when patients entered the initial study, and then certainly a safety follow-up period after the last completed dose. And so some key points of the study is that the primary outcome measure was changed from baseline MG-ADL score at week 26 in the combined study population. So both the acetylcholine receptor and MuSK autoantibody groups were pooled together. Some typical key eligibility criteria, these were patients with either acetylcholine receptor or MuSK autoantibody confirmed generalized myasthenia gravis with an MGFA classification between 2 and 4. Standard enrollment criteria used in other interventional clinical trials was an MG-ADL score of 6 or higher, QMG score of 11 or greater, and also some key design considerations that I wanted to point out here is that during the course of the clinical trial, after week 4 or at week 4, all participants in this study underwent a protocol-specified steroid taper down to 5 milligrams per day, and they all achieved that by week 24 of the study period. Again, this was a global study across 79 sites in 18 countries, so truly an international effort. And here are the study endpoints. So as I already mentioned, the change in ADL score between baseline and week 26 for the pooled population of antibody-positive disease. And there were a number of key secondary outcomes, and we say, why so many? We approached this in a hierarchical order to reduce multiplicity. And the first key secondary outcome was change in the QMG score at week 26 in the combined study population, followed by change in ADL score for acetylcholine receptor, a change in the QMG score again for acetylcholine receptor, and then similarly, the ADL and QMG score changes for the MuSK subpopulation. So I'll present those data here. So as already noted, we randomized a total of 238 patients, and these were 190 in the acetylcholine receptor autoantibody positive group and 48 in the MuSK autoantibody positive group. I will point out that this is the largest enrolled cohort of MuSK antibody-positive disease in a placebo-controlled clinical trial to date. Most of the patients actually completed through the controlled period. So for instance, we have a little over 90 to 95% in the acetylcholine receptor that completed, and 95 to 100% in the MuSK cohort in the placebo and inebilizumab arms. In terms of the study population, so the demographics and baseline disease characteristics were well balanced between the inebilizumab and placebo groups. We have a mean age in the 47 or so age. The population was comprised of predominantly women between 55 and 66%, again well matched. Disease duration was about 6 years. ADL score was about 9. QMG score was between 16 and 17. MGFA clinical classification, about 50%, a little over 50% of patients were MGFA class 3 or 4. MGFA class 2 accounted for, again, the other half of the population. So it was a split between mild versus moderate to severe disease. We also looked at the baseline steroid use. So not all patients were required to come in on steroids, but a large group of patients did in fact come in on steroids. And we looked at what was the baseline steroid dose. As you might recall, there is a forced or protocol specified taper. So 96 to 98% of patients were, greater than 80% of patients were on a prednisone dose of more than 5 milligrams at study entry. So this is our primary endpoint. There was a statistically significant change in the MG-ADL score in the combined study population at week 26, with a placebo group decreasing by 2.2 points on the ADL versus 4.2 score reduction in the inebilizumab group, and that met statistical significance. You can see that the trend is actually going towards separation between the groups after week 8 in the combined population here. Our key secondary outcome measure, which looked at QMG score change at week 26, again, showed a statistically significant difference between the two groups, placebo 2.3 versus 4.8 in the inebilizumab treated arm, and that was statistically significant. Looking at the MG-ADL for both AChR and MuSK patients, these were statistically significant. You can see the trend down. I will point out that while statistically significant, the confidence intervals that you see, they're greater, and that has to do mainly with the fact likely that this was a smaller group of patients as compared to the AChR group. We see similar trends for AChR and MuSK for both QMG. However, while the AChR group was statistically significant at week 26, the MuSK at week 26 was not statistically significant, but there was a trend. Again, high or great confidence intervals. The overall safety data, there was no increased safety events in the inebilizumab treated participants versus placebo. Similar percentage of participants with safety events in both AChR and MuSK groups, in case you were wondering. There were 3 deaths reported. All 3 were related in part likely to myasthenic crisis. So in conclusion, inebilizumab demonstrated clinically meaningful efficacy and a favorable safety profile in the combined population. In the first phase 3 study, which included a protocol-required steroid taper, the study findings support the role of anti-CD19 B-cell depletion therapy in myasthenia gravis. And inebilizumab, based on its mechanism of action, targets the upstream immunopathogenic disease drivers and represents a novel approach in the management of myasthenia gravis. No new safety signals were identified. And safety profile was very similar to an indication for NMOSD with this intervention. So our next steps are to publish the top-line results, meaning our primary manuscript, early next year. That's already underway. And data analysis of additional endpoints such as MG exacerbation, steroid tapering, quality of life, and durability of responses is ongoing. And additional assessments of safety continue to be ongoing per the protocol. Again, there's a lot of analyses that are underway. This is top line, fresh off the press at this point in time. So I thank you for your attention. And I will introduce our next speaker, which is Dr. Vu.
Good morning. On behalf of my co-investigator, it's my privilege to present to you these data on the safety and efficacy of BCMA-directed mRNA CAR T-cell therapy in generalized myasthenia gravis. Here are my disclosures. We still have a lot of unmet needs, particularly in the upstream therapy area, particularly B-cell directed therapy. So this is a study that looked at a way to reduce the cells that produce antibodies. And so the Descartes 08 mRNA cell therapy is different than the conventional CAR T cell therapy in the sense that we do not need lymphodepletion or chemotherapy before infusion. It can be administered on an outpatient basis, delivered at therapeutic level with subtoxic doses, and the PK and PD, the controllable, and the cell modification is transient with no genomic integration. So the cells that this therapy is directed toward are the plasma cells, the plasmablasts, and the plasmacytoid dendritic cells, which produce antibodies and also inflammatory responses. Descartes 8 is engineered by transfection of autologous CD8 T cells with mRNA encoding anti-BCMA chimeric antigen receptor. The treatment can be processed for infusion within about 3 weeks. And the data of today is from the phase 2B, comparing Descartes to placebo. So the eligibility criteria are pretty standard. And in contrast to other studies, complement inhibitors are allowed in this study. FcRn inhibitors was excluded, mainly because the available treatment on the market right now are given cyclically, and therefore we cannot control for the response to the treatment during the study. So after a successful screen, the patient underwent leukapheresis and was randomized 1-to-1 to either Descartes 08 or placebo. And both groups received 6 weekly treatment, and the endpoint was measured at month 3, after which unblinding occurs. And then the placebo-treated group now get Descartes infusion, and both groups was followed at various time points. The primary endpoint was the proportion of patients with MGC improvement of 5 or more points at month 3 relative to placebo. We also look at secondary measures, including safety and tolerability, and also the standard measures of efficacy, QMG, ADL, and quality of life. So 36 patients were enrolled. 31 of them was randomized, 18 to the treated group and 13 to placebo. And the intention to treat group consists of 26 patients. The 5 patients, so the intention to treat groups consists of patients who were seen at medical centers who qualified for the MG composite scores. But 5 patients were treated in community setting as a proof of concept to show that this treatment can be given in the community. So here are the baseline characteristics. More women was in the Descartes group, and the QMG was 17 in the Descartes group versus 15 in the placebo group, and the ADL was 10. In terms of MG antibody status, they were pretty well matched. In terms of previous treatment, again, because of small number, there was several imbalance as shown by the asterisks here. And you can see that more patients in the placebo group had complement inhibitors at baseline. Thymectomy was also more common in the placebo group. And then there's some disparity in also in the immunosuppressant treatment and the complement inhibitors. So here's the baseline. Here's the endpoint, primary endpoint. So In both the overall population and the AChR population, there was a statistically significant difference in the proportion of patients with MG composite scores of 5 or more at month 3. Looking at the overall population, we see statistical significant changes in the MG-ADL score and also in the MGC score. Looking at the AChR positive patient cohort, statistical significance was noted in the MG-ADL, MGC, and QMG. The placebo-treated group, the score reduction in measures of disease activity or the placebo effect was more robust in the seronegative group here versus those who had antibodies. At 6 months, we still saw a deep and durable response to treatment. In terms of safety, none of the patient had the cytokine release syndrome or the ICANS that you sometimes see with patient with traditional CAR T-cell therapy. Most of the AE was transient or mild. The only difference here is that patients who were treated with DCARDs had fever versus none in the placebo group. These fevers ran up to about 40 degrees. Most of them was in the 38, 39 range. In fact, all of them except for one. And the fever resolved within 24 to 48 hours. In terms of antibodies titer, you can see that in the treated group, there was a 15.8% decrease at month 3 versus a 38% increase in the placebo group at the same time period. And for the patient that we have data available, 3 of them went out to month 12 currently, and the deepest reduction was seen at month 9. DCARDS 8 does not deplete a broader antibodies repertoire or decrease vaccine titers for common viruses. And so in summary, 6 once-weekly doses of DCARDS 08 was associated with a significantly greater proportion of MGC responders than placebo at month 3. Responses was greater than clinically meaningful and was durable at last follow-up up to 6 months. The data does support outpatient administration with 1-hour post-infusion monitoring, and antibody level was reduced at month 3 and peak at month 9. The medication was not associated with an increased rate of infection, hypogammaglobulinemia, or reduction in vaccine antibodies titers. And so the data from this study will be tested further in a phase 3 randomized placebo-controlled trial, hopefully sometime next year. We would like to thank the patients, the caregivers, and the whole entire team for this effort. Thank you. I would like to introduce Dr. Julia Manison for the next presentation.
Thank you so much to the organizers of this meeting for selecting this abstract for an oral talk. My name is Julia Manison, and today I will discuss the preclinical pharmacology of S1117, which is a novel engineered Fc-fused IgG cleaving enzyme from Seismic Therapeutic. These are my disclosures. So as we have seen over the past few years, IgG-lowering therapy has shown significant efficacy in autoantibody-mediated diseases such as myasthenia gravis. And enzymatic IgG degradation offers a new therapeutic opportunity in this space. At Seismic Therapeutic, what we've done is we have identified and engineered S1117 from a highly specific, naturally occurring IgG bacterial protease called IDES, which derives from Streptococcus pyogenes. We have enhanced the protease by fusing it to a human FC domain to extend the half-life of the molecule. And we have also used our IMPACT machine learning platform to remove B and T cell epitopes in order to reduce the risk of immunogenicity. S1117 offers a multi-mechanistic approach to the treatment of diseases such as myasthenia gravis, which has the potential to improve on current therapeutic options as they stand now. S1117 cleaves IgG. It cleaves IgG antibodies at the hinge region, which separates the FAB'2 portion from the FC portion, and as a result, it reduces circulating IgG, and this reduction is rapid, deep, and sustained. It also cleaves IgG in immune complexes, as well as the B cell receptor in IgG-positive memory B cells. At the same time, S1117 directly reduces the ability of IgG antibodies to exert their effector functions, such as complement-dependent cytotoxicity and antibody-dependent cellular cytotoxicity. So I will go through some of these mechanisms in detail in the following slides. Using in vitro assays, we demonstrate that S1117 cleaves both soluble and immune complex forms of IgG. The figure at the top left shows the degradation of total IgG levels in healthy human plasma with increasing concentrations of S1117. S1117 cleaves all 4 subclasses of IgG. Similar results are seen in the plasma of myasthenia gravis patients, which is shown in the figure at the bottom left. And furthermore, as seen from the figure on the right, S1117 prevents immune complex-mediated interferon alpha production. The addition of S1117 results in lower levels of interferon alpha production compared to immune complexes that are generated from RNP and anti-RNP. Using both in vitro human and in vivo rabbit assays, we demonstrate that S1117 also cleaves IgG B cell receptor on memory B cells. The figure on the left shows the decreasing percentage of kappa-positive IgG-positive B cells in human whole blood with increasing concentrations of S1117, which results from the cleavage of the IgG BCR. The figure on the right shows increasing mean maximum % reduction of BCR MFI, representing the peak IgG BCR cleavage with increasing dose levels of S1117. When compared to a benchmark FcRn inhibitor, one of the therapies that's currently used in myasthenia gravis, S1117 demonstrates faster, deeper, and more sustained reduction of circulating IgG. And this we show in vivo. In this experiment, C57 BL6 mice received human IVIG at day -1, and then were subsequently injected with either a benchmark FcRn inhibitor or S1117 at day 1. Blood collections were performed at several time points to measure the reduction of human IVIG. Mice also received a second dose of IVIG at day 9 to measure sustained drug response. And as you can see from the figure on the right, a single dose of S1117, which is shown in the orange and the blue, achieved a rapid and deep reduction of human IVIG levels, and it maintained significant activity over time even after redosing IVIG. Whereas a single dose of benchmark FcRn inhibitor, shown in green here, achieved a less rapid, less magnitude reduction of human IVIG, which was less sustained, as you can see from the redosing of IVIG. As previously mentioned, S1117 directly reduces IgG effector function by separating the FAB'2 portion from the FC portion of the molecule. And here we demonstrate the capability in CDC and ADCC using in vitro assays. The top right figure demonstrates that increasing concentrations of S1117, which is shown in the turquoise color, reduce CDC, which is represented by increasing levels of cell viability. The reduction of CDC is even more effective than benchmark complement inhibitor, which is shown in orange, and as expected, FcRn inhibitor does not reduce CDC. We can see similar results at the bottom figure with ADCC as well. As we prepare to enter the clinic, we have projected S1117 pharmacokinetics and pharmacodynamics using QSP modeling and believe that we can achieve a titratable, convenient treatment regimen for patients, which is shown by some of the figures on the right. And here the figures demonstrate single dose projections, but we also have multiple dose projections as well. So for example, doses 2 and 3, which are in green and light blue, are predicted to achieve IgG reductions that have demonstrated therapeutic benefit in myasthenia gravis maintenance, but here we would achieve this with less than 1 mg per kg of drug administered about every 4 to 6 weeks. Dose 4 would be an example that is predicted to achieve a deep and a rapid reduction of IgG that can be applied in more acute situations such as myasthenia gravis crisis. So in this pretty rapid talk, I hope I've given you a glimpse of S1117, which is a novel engineered pan-IgG protease that demonstrates rapid and sustained reduction of all human IgG subtypes and addresses multiple orthogonal pathogenic mechanisms in autoimmunity with just a single molecule. It directly cleaves circulating, immune-complexed, membrane-bound, and BCR IgG without affecting other immunoglobulin isotypes. It is also expected to achieve dosing regimens that can be adjusted for both chronic and acute treatment and used as a treat-to-target approach, which may be titrated based on a patient's disease activity. It is being developed for subcutaneous self-administration, hopefully every 4 to 6 weeks for patient convenience. And finally, it will be entering the clinic pretty soon, and we hope to show more data as it comes out from the clinic. Before concluding this talk, I just want to acknowledge and thank the amazing Seismic team for all the wonderful work that has been done on this molecule, and as well as our wonderful collaborators on the work that has been done on the molecule and its relationship to myasthenia gravis. Thank you so much. And with that, I will introduce Dan Brennan to come in for the next talk.
Thank you, Julia. So we have a slight change in the schedule, and I would like to say good morning and thank you for inviting me and NMD Pharma to this session to present a bit about CLC1 inhibition in skeletal muscle, but also about our drug in development, NMD 670. Unfortunately, due to a series of travel and health-related challenges, our presenters and my colleagues from the medical and scientific team, Dr. Martin Sko and Dr. Vera Kiasova, are not going to be able to make it, so we're combining these presentations into one. Unfortunately, Dr. Kiasova was in the hospital all weekend in Spain, and Dr. Skov was flying from Denmark here to Savannah, and his plane turned around over Iceland, went back, was canceled, and he couldn't find another flight out. So here I am, and my name is Dan Brennan. I am a member of the executive team at NMD Pharma, and I am normally doing things like business strategy, corporate strategy, patient advocacy and engagement, and I'm wearing several hats, as we do, and quite happy to be here today to share this information that many of my colleagues have done over the years and are very proud about, and unfortunately just not here to share it. So NMD Pharma is a Danish biotech pharmaceutical company in Aarhus, Denmark, and it was founded 8 years ago by Thomas Holm Petersen, who is a holder of a PhD in physiology and previously was a professor of muscle physiology at Aarhus University. And much of his PhD work was focused in on ion channels found on skeletal muscles. Observations of neuromuscular transmission deficits seen in patients with various neuromuscular diseases such as myasthenia gravis, but also spinal muscular atrophy and Charcot-Marie tooth disease motivated Dr. Peterson to study chloride-1 ion channel, an ion channel that is specific to skeletal muscle and only found on skeletal muscle. He and the team have gone on to develop a platform of ion channel inhibitors designed with the goal of addressing skeletal muscle weakness, muscle fatigue, and lack of endurance felt by these patients in neuromuscular conditions. An entrepreneurial seed funding started out the company, followed by 8 years of work and $180 million in venture investment, which has led to the discovery, preclinical work, and development, initial clinical study, safety study, and proof of mechanism work completed on the chloride-1 ion channel inhibitor NMD 670, which is an oral, twice-a-day, skeletal muscle targeted treatment for patients with rare neuromuscular diseases. The chloride-1 ion channel plays a critical role in the proper functioning of skeletal muscle by maintaining electrical stability. And interestingly, the chloride-1 ion channel is found at the neuromuscular junction, but also along the entire muscle fiber and is highly concentrated around the T-tubules along the myofiber, with low evidence of impact from ongoing damage at the neuromuscular junction in myasthenia gravis. Its primary function is to regulate the flow of chloride ions across the muscle cell membrane, which influences the muscle's excitability and ability to contract efficiently. It stabilizes the resting membrane potential by counteracting the excitatory inputs from sodium and potassium ions movements. By doing so, it ensures that the muscle fibers do not become too easily excited, preventing spontaneous or involuntary contractions. CLC1 ion channels open after depolarization, allowing the chloride ions to flow into the muscle cell and help restore the negative resting potential in repolarization. In diseases highlighted by disrupted neuromuscular transmission, such as decrement or evidence of jitter and block from single fiber EMG, it was hypothesized that partial inhibition of this channel could have lower the threshold of activation and thereby amplify the muscle contraction, improve muscle strength, power, and endurance. This would be a new and unique drug target with the potential to provide improvements in muscle strength, power, and endurance across all skeletal muscles and a complementary therapy to other therapies that are used in these diseases. In healthy muscle where a motor nerve releases acetylcholine, a large increase in the end plate potential is observed, leading to a generation of an action potential. See the gray upper left-hand corner. And when several action potentials are required, the muscle is able to generate one each time it is needed, the gray lower panel. In myasthenia gravis, the end plate potential is reduced due to a lack of functional acetylcholine receptors and structural degradation at the neuromuscular junction, and that is the red upper panel. As a result, the muscle is no longer capable of generating all of the action potentials required of it, leading to reduced strength, weakness, and fatigue in the lower panel, with few or no action potentials generated after the first few. By inhibiting CLC1 selectively, NMD 670 is able to restore some of the amplitude of the end plate potential, the blue upper panel, and increase the likelihood of generating an action potential, restoring neuromuscular transmission, where action potentials come back as in the blue lower panel. On the next slide, we will see this translated into meaningful improvement in function. Similar to the previous slide, a muscle contraction in a healthy muscle should generate force reliably, as seen in the gray trace. In myasthenia gravis, the force generated is lower, and in this severe case, only in the first few hundred milliseconds during stimulation. By inhibiting CLC1, NMD 670 greatly improves the muscle force, the blue trace, and these changes here were observed within 10 minutes of dosing in a myasthenic rat. In addition, NMD670 was shown to be able to increase force from mild and severe levels of myasthenic symptoms in rats. This is the red force before NMD670, and the blue force is after NMD670 relative to 100% function. The effect is dose-dependent, or was dose-dependent, and onset was within 20 minutes and lasted for several hours. These findings encouraged a phase 1b/2a study where a single dose of NMD670 was administered and investigated 3 hours and 5 hours post-dose. And here is the design for this phase 2a proof of mechanism study, as we call it. There were 12 patients with mild symptoms of generalized myasthenia gravis. The average QMG score at baseline for these patients was about 9, and a standard deviation of 3.4, so relatively mild. And they received, in a crossover design, each patient received 1 dose of either 400 milligrams, 1,200 milligrams, or placebo at subsequent visits. Week 1, they came in, they got 400, 1,200, or placebo, then was measured on QMG at 3 hours and 5 hours, and then went home, it washed out, came back in for visit 2, received either dose 2 or placebo, and then finally in the 3rd week, received the final aspect. As mentioned, 3 hours after that dose, the QMG was performed, and again at 5 hours after the dose. In the study, NMD670 was seen to be safe and well tolerated. Again, this was only 1 dose at 3 different times in 1 day. The incidence of treatment-emergent adverse events were similar across treatment arms. No serious or severe treatment-emergent adverse events were reported, and we saw no excessive on-target myotonia effects at either the 400 or 1,200 milligrams, but there was some evidence of slowed hand release of an object at 1,200 milligrams in 1 patient. Sorry here. Time to maximum plasma concentration is 2 hours. Both half-life is about 5 hours, and concentration of NMD670 increased with increasing dose. On the bottom left chart, you can see the QMG data. On the days that patients received placebo, their symptoms worsened at 3 hours and further at 5 hours. You can see that in the red line. At 3 hours, gaining about a half a point, and at 5 hours, nearly a full point. 40 to 50% of patients on either 400 milligrams or 1,200 milligrams at 3 hours and 5 hours post-dose achieved a response is defined as 2-point improvement on QMG, and that's on the right-hand side. On the left-hand side, you can see the average improvements without a dose increasing effect by the NMD670 at both 400 and 1,200 milligrams, and this was statistically significant. While new and conventional existing therapies fight the immune system of generalized myasthenia gravis, many patients are left to deal with persistent life-altering symptoms. We believe that symptoms results from ongoing and lingering damage that is difficult to correct at the neuromuscular junction that continues to impair muscle function. And we're interested in studying whether or not NMD670 can continue to be added to the current standard of care treatment, be safe and well tolerated, and alleviate the burden of persistent and fluctuating symptoms by providing muscle strength, endurance, and power. And so now we have moved into the clinic, a phase 2b study, and it is called the Synapse MG dose ranging and dose finding study, which is a double-blind, placebo-controlled study of NMD670 in patients experiencing persistent symptoms or fluctuating symptoms despite ongoing stable treatment with conventional immunosuppressants or immunomodulatory treatment. The objective is to assess the safety, tolerability, and efficacy for 21 days of dosing with NMD670 in adult patients with MG. And you see here some of the primary endpoint will again be the QMG total score with secondary endpoints of MG-ADL, the composite score, MG-QOL, neuroqual fatigue, and we have an enriched statistical design, which is a multiple comparison procedure, which allows us to compare with fewer patients, not necessarily each group against each other, but against the entire dose response curve. Here are the sites. A total of 38 clinical sites are desired to be brought on board across 10 countries. We currently have these number of sites that you see here listed in a variety of different countries across the US and Europe. And my last slide here, with a misspelling it seems, is our trial sites here in the US. In yellow are the ones that have been signed and contracted and are already enrolling patients. We had our first patient enrolled in the summer, and we hope to get the remaining sites up and running and screening and enrolling patients here soon. So I'd like to thank you very much for your time here and allowing me to step in and present for my colleagues. And also wanted to say that this is our study design in generalized myasthenia gravis. We also have 2 phase 2 studies in other diseases, one for adults with spinal muscular atrophy, and also in a variety of subtypes of Charcot-Marie tooth disease. Both studies are up and running, and we believe that they also are demonstrating an effect of a weakened nerve signal and demonstration of single fiber EMG decrement or deficits or decrement in neuromuscular NMS, but also the ongoing denervation and reinnervation within those disease states is contributing to a weaker signal at the neuromuscular junction. Thank you very much for your time. And now, I'd like to present the next speaker, who is Dr. Aidan, hold on a second, Haghikia, Dr. Aidan Haghikia, thank you.
Hello, thank you very much. My name is James Chung. I'm the Chief Medical Officer at Carverna, cell therapy company founded in 2018 focused exclusively on autoimmune diseases. Like the previous speaker, Professor Haghikia couldn't make it today because of an urgent issue that was at his university. So I'm speaking in his place for the study team. Professor Haghikia is the director at the Department of Neurology at the Medical Center, University of Hanover in Germany. And I'll show you some of the work that he did to really open this field for CD19 CAR T therapy in autoimmune diseases. So I'll be talking about our phase 2 study with Carverna 101, which is an anti-CD19 CAR T therapy. It's an autologous, fully human CD19 CAR T therapy for refractory generalized myasthenia gravis. So of course, myasthenia gravis is an autoimmune disease with great degree of disability and even mortality. And we had heard a really wonderful talk to open up this meeting today about the role of autoantibodies and the various ways in which they have pathogenic effects. Treatments, of course, are really directed at the autoantibodies and other factors involved in damage at the neuromuscular junction. And recent therapies are going really upstream at the source of these autoantibodies, and that is the B cells. And we saw a presentation about the monoclonal antibodies and its impact, starting from the CD20 antibodies to the CD19 antibodies, which have a broader range of B cell subsets which they impact. And while they're effective, what we see is that they deplete peripheral B cells very well, but there are a lot of pathogenic B cells that are in the tissue, and the tissue-resident B cells are the source of much of the pathogenic autoantibodies. And we know from the oncology area that CD19 CAR T cells are quite effective at depleting B cells, not only in the periphery, but in the deep tissues. And it's very successful in the B cell malignancy area. And as you know, it's recently been applied more in the autoimmune disease setting, where what some recent studies have done with lymph node biopsies after CD19 CAR T therapy compared to monoclonal antibodies, and this is work done by Georg Schett in Germany, is that not only are the B cells more completely depleted in the tissues, there is a disruption of the B cell follicles where the interaction of B cells and T cells occur. And this really offers the promise of an immunologic reset that not only gives you symptomatic relief with chronic administration of your therapeutic, but a single dose administration which may lead to long-term drug-free remission. And that is the promise of the CD19 CAR T therapy. Carverna 101 has a construct that was licensed from the NIH by Dr. James Kochenderfer, who designed Yescarta, which is a very successful CD19 CAR T therapy in the B-cell malignancy area. And he actually designed it with safety in mind from the get-go. So he retained the CD28 co-stimulatory molecule from Yescarta. It changed some other components like the hinge and transmembrane domain to CD8 alpha, and he swapped out the murine CD19 binding domain with a fully human one. And what he found that was in vitro testing showed that it produced lower cytokines compared to Yescarta, and he reasoned that this may lead to a safer profile, and did a study with 20 patients with B-cell malignancies. And he found that compared to Yescarta, it did indeed produce lower cytokines in the patients, and that was associated with a much reduced neurotoxicity profile and a 10-fold decrease in severe neurotoxicity compared to Yescarta. So in addition to the lower immunogenicity with a fully human construct, lower cytokine production, and a safer profile, these properties we felt was really optimized for use in autoimmune diseases. And so the very first patient that we treated with Carverna 101 in autoimmunity was by Professor Haviki and his colleague Dimitrios Mougiakakos in Magdeburg. As a patient with severe refractory anti-acetylcholine receptor seropositive myasthenia gravis. She was 33 years old, had been treated with multiple agents, with rituximab, bortezomib, CellStab, you can see, and really was deteriorating. She was having multiple myasthenic crises. It was very difficult to control with rising titers of her autoantibodies. And she was treated with Carverna 101. And you can see that, first of all, the results were rapid and the results were dramatic. You can see on the left-hand side the decrease in autoantibody titers with about 70% decrease by 2 months, which continued to decrease over time. Very importantly, the measurement of anti-vaccine antibody titers show that that did not change. That's consistent with the CD19 target that doesn't extend into the long-lived plasma cells. And these are the kinds of findings that we're seeing in the other diseases that are studied with CD19 CAR T therapies as well. You can see that her walking distance, and she required a walker prior to the CAR T therapy, very rapidly she was able to be free of her walker and to be walking longer distances without assistance. You can see her arm holding time improving rapidly over time, and her more composite measures of myasthenia gravis improving as well, and her myasthenia crises have stopped. And importantly, she was able to wean off her steroids and to be treatment-free. And you can see here on the left-hand side that just about a week after treatment, she's able to walk, but she has needs assistance in walking, whereas just within 2-week period of time, she's free of her walker, and she continued to improve even more over time. The second patient is 75 years old. We thought it was important to also include patients who are older and have elderly onset myasthenia gravis. And this is a patient who was diagnosed more recently in 2022, but had a very rapid and severe course, had severe swallowing difficulties, had to have a PEG tube for feeding, and really had run out of options after IVIG, rituximab, and was deteriorating rapidly, and was treated with Kyverna 101, and he also made a very rapid recovery, so that by 6 months post-CAR T therapy, he was back doing what he loved doing best, which is gardening in his backyard. So a really good outcome for him. What's true about both of these patients, and this, as I mentioned, is really the promise of CAR T therapy, is that they received a single therapy or single dose administration of Kyverna 101 after a very refractory course, and had really durable responses. First patient is now actually beyond 16 months, and the first patient is out 12 months, both drug-free and still doing quite well. The B cells have repopulated. The first patient, we're still waiting for the B cells to repopulate. For the second patient, the B cells do come back probably a little earlier than what you see in the B cell malignancy setting, likely due to the more normal bone marrow, but just to say that the B cells come back, but the disease remains under control. And so the phase 2 study was started after seeing these very promising results. As I mentioned, I think some of the results with monoclonal antibodies, such as rituximab, the data which were available at the time we started the study, it seemed a little mixed, but given the very dramatic results, we were able to start with a target dose of 100 million cells, and starting at phase 2, it has an RMAT designation. And for CAR T therapy, you screen the patients, you apheresis them, and then the cells are prepared and manufactured. About a week prior to the CAR T administration, there is lymphodepletion with cyclophosphamide and fludarabine. That is to allow the cells to be able to proliferate and expand. And then you give the infusion, and you're in the hospital, in this case for 10 days for close monitoring. There is CAR toxicity, such as cytokine release syndrome and neurotoxicity, ICANS, but they're much less severe than what you see in the oncology setting, but you do still see them. The first patient actually had no CRS or ICANS. The second patient had grade 2 CRS. After 10 days, they leave the hospital. They're monitored for the first month near the hospital, but still outside of the hospital, and they're followed for the study for 2 years. The primary endpoint is something you're very familiar with, MG-ADL, 24 weeks. We're also looking closely at safety, of course. And the secondary endpoints are QMG and MGC scores at 12, 24, and 52 weeks. We'll, of course, monitor the autoantibodies and a variety of biomarkers that'll give us an indication of the pharmacodynamic effect. We're very interested in what an immune reset may look like for these patients over time. For key inclusion criteria, 18 to 75 years. As I mentioned, we wanted to make sure that we got the full spectrum of the age range to get that bimodal age distribution of the different patient populations there. Class 2B to 4 for the MGFA criteria. Patients who are seropositive, although we do recognize that there are seronegative patients who may benefit with an MG-ADL score of greater than or equal to 6. And they have to have failed at least 2 immunosuppressants or 1 and are IVIG dependent. And we have some key exclusion criteria as well, prior cell therapies or hematopoietic stem cell transplant. So Kyverna 101 is a novel therapeutic approach. We believe that really has the potential to transform the way myasthenia gravis, but also other autoimmune diseases are treated due to its deep tissue penetration and disruption of the immune system at that level to lead to longer remissions without administering concomitant medications. So we have both, as I mentioned, fast track and regenerative medicine advanced therapies designation for myasthenia gravis, as well as for stiff person syndrome. And we have this phase 2 trial both in the US as well as in Germany. And not only SPS, but we also have a phase 2 trial in multiple sclerosis. Thank you very much. So I think the next speaker is Dr. Vu.
Well, this feels like deja vu. again, it's my honor to present to you the top-line results from the double-blind placebo-controlled randomized phase 3 Vacity study on behalf of my co-investigators. This study is sponsored by Janssen, and here are my disclosure again. Nipocalimab is a monoclonal antibody designed as a FcRn blocker. The inclusion criteria for this study are pretty standard. Both seropositive patients, including AChR, MuSK, and LRP4 antibodies are included, and some seronegative patient as well. The patient was randomized 1-to-1 to nipocalimab or placebo, and they receive a loading dose at baseline followed by IV infusion every 2 weeks. And then after a 24 weeks period, they were able to enter the open label study. So in terms of treatment disposition, 98 patients were in each group. 89% finished the study in the nipocalimab group and 84% in the placebo group. And the reason for discontinuation are listed below. 2 patients in the placebo group died both from heart disease. 1 patient in the nipocalimab died due to MG crisis. None of these deaths were considered related to IP. In terms of demographics, pretty well balanced. Slightly more patient in the nipocalimab group was women. The QMG score was 15, and the ADL score was roughly 9. In terms of antibody status, more patients in the placebo group had AChR antibodies, and more patients with MuSK was in the nipocalimab group. So here's the primary endpoint, which was defined as the Myasthenia Gravis, MG-ADL score changed from baseline to the average over week 22, 23, and 24. And nipocalimab plus standard of care demonstrated a statistically significant change in MG-ADL of 4.7 versus 3.25 in the placebo group with a p-value of 0.002. Similar result was seen in the QMG, and the change there was 4.86 versus 2.05 in placebo, again, statistically significant. In terms of other key secondary endpoints, the responder rate from baseline to week 22 to 24 was 69% versus 53%, and there was statistically significant there as well. The next secondary endpoint was the early response rate, defined as that happening between week 1 or 2, and that was not statistically significant, and due to hierarchical testing to account for multiplicity, the next 2 key secondary endpoints were not tested, but you do see a numerical difference between response from week 4 to 24, testing for durability, and testing for depth of response by 50 or more % improvement from week 22 to 24. Of note, about a third of the patient in the nipocalimab group versus 13% in the placebo group achieve MSE at any point, and 10% of the nipocalimab group achieve MSE versus 1.3 placebo for more than 75% of their time during the double-blind phase. In terms of subgroup analysis, a patient with AChR and MuSK antibody had statistically significant primary endpoint. The seronegative patients did not show a difference between the 2 groups. In terms of biomarkers, the total IgG level was decreased by about 69%, And for anti-AChR antibody level, there was a 65% decrease versus 10% in the placebo group. And for the MuSK patients, 39% versus 4.4% decrease. In terms of safety and tolerability, the drug was pretty well tolerated, and there was no difference other than there were more patients with muscle spasm in the treated group, and there was peripheral edema in 11% of the patients in the treated group versus none in the placebo. Cholesterol level was increased, including HDL and LDL, but the cholesterol to HDL level ratio was less than 4 at week 24, and there was no cardiac side effects. So in conclusion, Vacity was the first registrational study of an FcRn blocker to show sustained efficacy through 6 months of fixed schedule dosing in a broad anti-positive population. It demonstrated statistically significant and meaningful improvement in MG-ADL and QMG, and the percentage of responders was also higher. More patients achieved sustained response from week 4 to 24 with nipocalimab compared to placebo, and more patients had greater than 50% improvement in MG-ADL with nipocalimab. And overall, the drug was very well tolerated. Thank you so much for your attention. I would like to introduce the next speaker, Dr. Jonathan Strober.
Good morning. I'm honored and excited to now present the safety and effectiveness of nipocalimab in adolescent patients in an open-label phase 2/3 study. My disclosures. You've already just seen this slide, so you've heard a little bit of what nipocalimab does. So the objective of this study was to evaluate the pharmacodynamics, pharmacokinetics, efficacy, and safety of nipocalimab in pediatric patients with generalized myasthenia who had insufficient clinical response to ongoing stable standard of care therapy. And just to note, these were 12 to 18-year-old patients, and the cutoff for the study for the data was December 15th in 23, so almost a year ago. So patients in this cohort were 12 to 18 years of age. They had to be had generalized myasthenia class 2 to 4, seropositive for acetylcholine receptor or anti-MuSK antibodies, and have suboptimal response to current therapies as determined by the investigator. They were screened. 7 patients were screened. 5 entered the active treatment at the time of this cutoff, and where they received nipocalimab 30 mg per kg, loading dose followed by 15 milligrams per kilogram every 2 weeks for 24 weeks. And then after that, they all entered the long-term extension phase where they were able to get either the 15 milligram per kilogram dose every 2 weeks plus standard of care, or nipocalimab 30 milligrams per kilogram every 4 weeks. The primary endpoint was the effect of nipocalimab on total serum immunoglobulin G and safety and tolerability, with looking at functional data, the MG-ADL and QMG. So these patients age range 12 to 16 with a mean of 14, mostly female, and a higher percentage of Asian patients. The baseline MG-ADL was on the lower side, 4.29, with a range of 2.5 to 9.5, and the mean QMG of 12.5. Patients had a mean duration of disease for 4+ years, ranging from 0.8 to 11.5, with onset of disease from 6 months to 13 years. Half of the patients, or just a little over half the patients, were classified as 2A, and then there was 3 that were classified as 3. And then 7 patients, all of them were on concomitant medications, as you expect, immunosuppressants being the most common, corticosteroids, and then other drugs as well. There we go. So the primary endpoint was to look at total serum IgG, and this was conducted in 5 participants who had actually received 1 or more doses of nipocalimab and had 1 or more post-infusion samples evaluable for serum IgG. And you can see, kind of similar to what we saw in adults, there was about almost a 70% drop in serum IgG in week 2, and then this persisted out through week 24. Try that. All right. Looking at the secondary endpoints, there was a clinically meaningful reduction in the MG-ADL score starting at week 4 and maintained through week 24. The score initially was 4.29 in mean and improved by a little over 2 by week 24 in these patients. 4 out of the 5 participants actually showed 0 or 1 MG-ADL score at week 24. Looking at the QMG, again, we saw a drop in the QMG going from a mean of 12.5 and improved by almost 4 points by week 24, and this was sustained. It was generally well tolerated, and there were no significant adverse events. There were no adverse events leading to discontinuation or adverse events of special interest through week 24 in the patients. And the most common adverse event was nasopharyngitis. So in conclusion, the primary endpoint, efficacy, nipocalimab, the 30 milligram per kilogram loading dose followed by the 15 milligrams Q2 weeks, showed a statistically significant reduction in total IgG in adolescents with generalized myasthenia. There was a clinical meaningful reduction of MG-ADL and QMG scores at week 4 and maintained through the 24 weeks. And it was safe and tolerated in adolescent patients in the study. And so this is actually the first clinical trial data reported for an FcRn blocker in adolescents. It's an ongoing trial. We're now enrolling younger kids in the US down to 8, I think down to 2 globally. And I think I helped catch us up a little bit on some time there. Okay. Thank you.
Since we're a little bit behind, let us just proceed to the next section, which is Introduction to Patient Care and Hot Topics. It's my honor to introduce to you the next speaker, Dr. Howard. I met Chip in 2013, and he's been my teacher in MG-related matters since then. I consider him the father of modern MG therapy, and it's such an honor to have him here to talk about medical education for healthcare professionals. Thank you.
Thank you, Tuan. Thanks to the organizers for the invitation to speak. We're going to change gears, and we're going to talk about education. And we're going to talk specifically about the needs-driven medical education for healthcare professionals in terms of myasthenia gravis. And I want you to think back 10, 15 years. How did you learn about MG? Was it your mentor? Was it through reading, etc.? And I think you'll find it was pretty lacking. These are our conflicts, speaking on behalf of this team. So in 2020, there was a recognition that the rapid change in the healthcare landscape for MG required that we rethink our educational principles. An extensive literature search was done between 2009 and 2019, and there was little existing research and information on learning needs as well as professional practice gaps in MG. And so it was decided that we needed to take a comprehensive approach and to cultivate an international MG community, provide a form of knowledge where we could share, we could learn, and interact with each other based on outcomes which was the focus. And so a learning needs assessment was performed, and it was very interesting. The group went back to the educators and said, how do you do this? How do you design learning programs that would be impactful? And the key to it, using this logic model, is you start at the end and you work your way backwards. So what do we want the impact to be? What are the outcomes we're going to use? What metrics do we need to assess that? Then what are the activities? And then what are our resources and the inputs? And having done that, you then build it out forward. And it's something I knew nothing about. And this was all in the realm of the educators. And so a 10-year ambition was designed. And the key was we wanted lifelong transformational learning. We wanted to cultivate this global community of practice and then improve the lives of our patients and families with myasthenia. We put together an international steering committee that's shown here. A few of us from the States, Renato from Milano, Kimiaki from Japan, and Heinz from Germany, and then developed initially this Rare Disease Connect module, if you will, where the first 1 to 2 years focusing on MG experts, and then from year 3 and beyond, as you will see, we started to branch out to include others within our MG community, patient advocacy groups, nurses, patients themselves. And we created touch points across the first 2 years. And based on our predefined outcomes that we wanted to cover immunology and pathophysiology, disease burden, as well as treatment burdens, the classification of disease, and you can see the list here. What are our challenging populations? What interprofessional collaboration can we do, and where's innovation going? And this is sort of the timeline of where we've been and where we're going. We started in 2021 with a 3-day virtual meeting in the midst of COVID. We had 127 HCPs involved. And then the following year in 2022, we met in Madrid, Spain for a 2-day meeting. And subsequently last year or this year in Berlin. And we're meeting in a few weeks in Amsterdam. In addition to that, we've created a virtual online community. And you can see those numbers in red. And it's growing, up 70% since 2021. And I would urge you to join our online community. And I have a brochure here in the front row that you can take. And with this online community, we're looking at case study videos. We're looking at panel discussions, infographic series, webinars. And this is all available to you, e-learning modules, for instance. And because of difficulty in transporting folks from states to Europe, et cetera, we are creating a US version, satellite, if you will, of this called USRDCN's annual summit that will meet at the time of the American Academy of Neurology meeting on the West Coast this forthcoming April. So we've achieved success, I think. And these are just some of the comments and the feedback that we've received. Unprecedented activity. Overall, I'll come back next year. Improved my knowledge. And so I think we've been successful in the short time we've gotten this thing going. And so we believe we are providing invaluable feedback. We've generated this community of healthcare individuals of all levels to be involved with this. So in summary, I think this program supports robust education in MG. It's product agnostic. It's truly an educational program. It's shaped by those of us in the MG community, for the MG community, using robust educational design principles. It's inclusive in terms of physicians, nurses, advocacy groups, foundations, patients. We're more than 500 participants in our global virtual community. And again, I urge you to seek us out and get involved. The net result is that we improve the care of our patients and their families who have MG. So it's invaluable as far as I'm concerned. It's interprofessional, as I said, in terms of education, multidisciplinary, and we'll be having forthcoming meetings here in the US as well. Thank you very much. And I'd like to introduce Kelly Gwathmey from the Medical College of Virginia. She'll be our next speaker.
All right, good morning, everyone. Thank you so much for the opportunity to present on behalf of our collaborators on the Myasthenia Gravis Foundation of America Global MG Patient Registry, Characteristics, Insights, and Learnings After a Decade from 2013 to 2023. Here are my disclosures. And our objectives for the next few moments are to briefly describe the MGFA Global MG Patient Registry and to report the baseline demographics and disease characteristics of this registry. So this is, as many people in this room know, a self-reported survey of patients with myasthenia gravis that was established in 2011. The first participants were enrolled in 2013, and participants are surveyed about twice a year, summer and winter time. So as I will show you in just a moment, there are over 3,500 participants. And if you assume that we have about 78,000 MG patients in the US, that's about 4.5% of the MG patients in the US have participated in the MGFA Global MG Patient Registry. To compare this to the type 1 diabetes registry, where about 14,000 are participating out of 2 million patients with type 1 diabetes in the US, that's only 0.7%, so we think we're doing pretty well. So the MGFA Global MG Patient Registry is powered by Health Storylines, which is a platform by Alira Health. And so it characterizes the natural history of disease and its impact on patients over time. These surveys are directly administered to the patients, and the enrollment is open, and longitudinal follow-up is open-ended. So participants can stay in the registry for many years, which is one of its strengths. Subjects are 18 years or older at time of enrollment and need to have self-reported myasthenia gravis. And so we do have descriptive analyses conducted on key clinical features and variables, which I'll show you momentarily. And then, as I already stated, these enrolled participants are contacted biannually to provide updates. And so the data I'm going to present to you is from July 2013 through June 2023. And we have patient-reported data, age, gender, sex, race, ethnicity, the state in which they live, their weight, employment status, and income status. And I'm not going to show you all of this data in the next few moments. We have patient-reported outcome measures, including the MG-ADL and QOL-15R. We have clinical and treatment characteristics as well, including antibody status, their medical comorbidities, exacerbations, thymic history, thymectomy, and also treatment history, as well as healthcare resource utilization, including overnight hospitalizations, ICU stays, and emergency department visits. So this is a distribution of our participants, again, through summer of 2013. You'll see the dark orange is 328, which is Florida, our most popular state from which our participants are coming, followed by Texas and California. So in terms of the demographics, just under 40% are men and just over 60% are women, for a total of 3,556. Nearly 95% are not Hispanic. And then in terms of age at time of diagnosis, for the men, 58 years, and for the women, just under 44 years. And then separately, age at time of enrollment, just under 63 years for men and just over 51 years for women. So looking at the antibody serostatus classification, you'll notice that the sample size is a bit lower at just over 1,800, and that's because we included participants who were confident, based on a series of questioning, on what their antibody status was. And so we have 62.8% that are AChR positive, 31.6% seronegative, 5.2% MuSK positive, and just a 0.4% that were LRP4 positive. Looking at employment status, you see a much higher number here, 3,527. Only 34% were full-time at time of enrollment, and then 9.9% part-time, and the rest unemployed at 56%. So this is a really beautiful figure. Age at diagnosis for the MG patients stratified by sex. So our women or females are in red, and then our men are in blue. And so you see that bimodal peak where women will peak in terms of prevalence before the men do. Looking at the distribution of treatments and therapies taken by our MG patients, and again, remember that this data cut was June 2023, that pyridostigmine and prednisone by far and away were the most commonly used treatments, followed by IVIG, mycophenolate, and azathioprine. So some of our newer therapeutics, such as efgartigimod and Ultomiris, are at the bottom just because they hadn't been around as long at the time that we did this data cut. In terms of comorbidities, you'll see that hypertension and hyperlipidemia were by far the most common, followed very importantly by depression and anxiety. So what we can also look at with the registry data is the MG-ADL score as an example of a patient-reported outcome measure that we can use. And so for the MG-ADL score across all of the participants, the average in the men was 5 and 7 in the women. And we can also stratify this data based off the antibody status. And so for AChR positive, for example, the average was 6. LRP4, again, very small numbers, only 8 participants, was 8.5, and MuSK was 7. Seronegative was higher at 8. So the take-home points here are that this is a decade's worth of MGFA global MG patient registry data that describes the baseline characteristics of over 3,500 patients with MG. The age of diagnosis was stratified by sex and serostatus, comorbidities, prior and current treatments also described. We can capture employment status. The distribution of the MG-ADL scores we can also look at by sex and antibody status at time of enrollment. These findings support the value and ongoing utility of this large-scale patient-reported registry, and the manuscript is in preparation and nearly ready to be submitted. And so with that, here is a QR code if you'd like to learn more about the MGFA Global MG Patient Registry. And I would like to introduce my medical student, Chris Evans, to present next. Thank you for your attention.
Hello, everyone. I want to thank the organizers for having me here. I also want to thank the fellow members of my team, especially Dr. Kelly Gwathmey, who's my mentor on this project. So our study is the common factors that influence fatigue in myasthenia gravis. I have no disclosures. As we know, fatigue is a really common complaint in myasthenia patients, but poses many challenges in terms of diagnosis and treatment. Specifically, fatigue can be separated and discerned into 2 separate kinds of fatigue. We have muscle fatigability, which is characterized by increasing fatigue with exercise. But we also have a central fatigue, which is more to deal with perceived fatigue and a lack of energy. So what do we know about fatigue so far? Fatigue in myasthenia, it's very highly prevalent, and it does not always track with disease control. In myasthenia, fatigue appears to be influenced by depression, sleepiness, and disease severity. And the worse the fatigue in myasthenia, the greater the impact on quality of life. And we also know from several phase 3 trials that fatigue in can be improved with treatment of myasthenia. We also know that fatigue is multifactorial. So just to give an example of that, myasthenia can lead to physical inactivity. Physical inactivity, of course, can then lead to muscle wasting, which of course can feed back into physical inactivity. What we want to look at in this study is the comorbidities that feed into fatigue. So those are our objectives. We wanted to study seropositive and seronegative myasthenia patients and correlate that with disease severity. We wanted to explore other factors associated with fatigue in myasthenia patients, and specifically we wanted to identify those treatable drivers of fatigue, and which can help guide providers in alleviating those symptoms. So our methodology, we had 46 myasthenia patients enrolled. They were both ocular and generalized, and the eligibility criterion was that merely that they had myasthenia and they endorsed fatigue. The labs we collected are a classic panel of labs to help assess fatigue: CBC, B12, vitamin D, TSH, testosterone. And you can see on the right the various questionnaires we posed to our patients to help quantify their disease severity, their level of fatigue, their level of depression, their level of sleep quality and anxiety. And so the demographics of our patients, 73% of them were female, roughly 80% of them were AChR positive. Their symptom and disease severity was relatively mild to moderate. And I just want to point out one specific element, which is the current symptom distribution. So as you can see there, roughly 83% of our patients had some manifestation at enrollment of disease symptoms. What that meant was that 8 of our patients were in remission in terms of myasthenia symptoms, yet they still endorsed fatigue, which really went to show that we had comorbidities driving fatigue within our patients. Here are some correlations that we found. Older age was protective of anxiety, depression, and fatigue. Lower vitamin D levels were associated with greater fatigue and depression, and lower vitamin B12 levels were associated with greater fatigue, and sleepiness was also associated with greater anxiety. Now, feeding this into a multiple regression model where we started controlling for age, disease distribution, sex, and disease severity, we found some very interesting findings. So on the left, I want to point out midway on the graph is myasthenia ADL total. And you can see there that for every 1 point increase in myasthenia ADL disease severity, we got a 2.2 increase in the neuro quality of life fatigue score, and a 1.48 increase in the fatigue severity scale. So we can see that disease severity is a driver of fatigue. We also see there right below it PSQI, a measure of sleepiness, was also associated with fatigue. But the key point I want to bring here is right below that, B12 deficiency. We found that B12 deficiency amongst our patients led to a 6.06 increase in the neuro quality of life fatigue scale and a 14.4 increase in the fatigue severity scale amongst our patients. Moving on, we also took a look at depression and anxiety in our multiple regression model. And what we can see here is, first off, just to point out that the male sex was actually protective of fatigue, against fatigue. But moreover, we found that depression was a significant driver of fatigue, and that once we started analyzing for depression, sleepiness actually dropped off as a driver of fatigue. And so we could see that depression was a driver both of sleepiness and fatigue. And anxiety, a couple dogs that did not bark in the study was that anxiety, while associated with it, with fatigue, didn't actually meaningfully correlate. Likewise, vitamin D, although it was associated with fatigue, was not statistically significant. So overall conclusions, fatigue is more commonly encountered in patients who are younger, female, and have more disease activity. Fatigue is associated with depression and less so with anxiety. And fatigue was still present despite accounting for disease severity, mental health concerns in our modeling, and that suggests those other comorbidities. So our conclusions, upon testing of additional causes of fatigue, we found that vitamin B12 deficiency was highly associated with fatigue. And vitamin D deficiency was clearly associated with fatigue, but it did not produce a statistically significant finding when we controlled for other variables. And sleepiness dropped out of fatigue findings once we considered depression. So our overall recommendations is that in fatigued myasthenia patients, we recommend screening for depression, vitamin B12 deficiency, and vitamin D deficiency. These are treatable drivers of fatigue. We also know that fatigue is tightly linked with disease severity, and those recent phase 3 trials show that treating disease severity can help improve fatigue. Our next steps, we'd like to do a larger prospective study looking at which factors are causative and not just associated with fatigue. And we would also like to explore the association of medicines, such as corticosteroids, with fatigue. We attempted to do that in the study, but the number of patients we enrolled on those medications was not large enough. Thank you. Here are my references. And I would just like to introduce Dr. Presnor next. Thank you.
Thank you, Christopher. That was a good segue into my study here. I'm going to be talking about safety and tolerability of whole body electrical stimulation exercise in adults with MG, a preliminary analysis here. So these are my disclosures. None of that will be used in this study. So just going over the background, as Christopher had went through all of these, most of these, I would say, is, as we know, it's an autoimmune condition. MG is affecting the postsynaptic membrane of the neuromuscular junction. And there's decreased availability of the acetylcholine receptor in fatigable muscle weakness. Maybe that's what causes the fatigable muscle weakness. We think maybe it's some dysfunction of the neuromuscular junction is causing the fatigue in these patients, which impacts the daily lives of the patients. And as Christopher mentioned, like it causes atrophy and deconditioning, leading to more comorbidities in these patients. Exercise can improve. What we think is exercise can improve physical function in MG, but traditional exercise paradigms are very difficult because, again, these patients are weak, MG patients are weak, and so there's some limiting the benefits that patients can experience. So what we are looking at is a whole body exercise, muscle stimulation plus exercise together promotes low impact exercise without external resistance while more fully activating the muscles, the motor units, and enhance the physiologic impact. So that's what we are looking at with the study. And the whole body electrical muscle stimulation exercise is actually FDA approved for normal people and young patients, the young healthy adults. This has been already approved. So we are trying to see if this is, how does this affect in myasthenia gravis patients? So that's what we were looking at. So that's the main objective of our study. So the objective is to assess the safety and tolerability of the whole body electrical muscle stimulation and to identify the effects of this on perceived fatigue of the patient. As Christopher already mentioned, perceived fatigue is what the patients feel, but fatigability is what you can actually record or actually measure, on the electrical stimulation, on, our testing in patients with GMG. So, and also examine the neuromuscular transmission and motor unit activity after this exercise study. So what is the study? It is a pilot study that it's an open label, so we wanted to enroll patients with myasthenia gravis, basically with the diagnosis. We did not do any other further testing. This was the investigator saying that this MG, it is a two-center study with Kansas City. This was funded by Kansas City Consortium for Musculoskeletal Disease. And basically, the patients should be able to stand for 15 minutes at least, and they should be able to do mild to moderate fatigability on arm, leg outstretched on QMG testing. And then major exclusion criteria is they should not have any pacemaker or any contraindications to using the electrical stimulation. And the study is basically 2 sessions per week for 4 weeks. They wear this body suit, which gives a little stimulations, and it's mainly in the areas that we would think mostly affected by myasthenia gravis, like the proximal muscles, the paraspinal muscles, So this is 2 sessions per week, 20 minutes, and this is a repetitions of 10 to 12 exercises along with the electrical stimulation. And this is using the device Catalyst fitness system. It also has an iPad associated with that, where they can adjust the stimulation as well as adjust the level of exercises and gives you an information on what exercises to do. And outcome measures, we are looking at safety and efficacy, mainly because this is the first time we are using in MG patients. And also we are looking at rating of perceived exertion and tolerability, whether this is tolerable for patients with MG. Efficacy, we are looking at fatigue severity scale, and we also are doing single fiber EMG before and after. And this is not in a muscle that we would normally do where there's not much of fatigue. We are looking at more the vastus lateralis, like in the quads, in the leg, where patients complain more of fatigue in the legs. And quantitative QMG testing and 6-minute walk test, and arm movement test is the other test that we are doing. So looking at the results, so what are the results? I mean, again, we have enrolled only 4 patients up till now, but we are still in the process of enrolling. And among those 4 patients, we looked at the visits attended, like the 32 visits, all the 32 visits were attended by the 4 patients. And visits completed, this was the completed, was 31 out of 32. 1 patient had problems with autonomic dysautonomia, and so there were some issues about some of the visits completed. But looking at the numeric pain rating scale in session, most of them tolerated it pretty well. And as you can see here, 53%. 1 patient had foot cramping, and that's what restricted from, the pain scale was mainly associated with that. And looking at the vital signs, we looked at what the blood pressure and heart rate response is, and most of them did well. There's not much change. There was only 1 instance of abnormal blood pressure and heart rate, and 2 instances of post-exercise, some drop in blood pressure in 1 participant. And mild to moderate rating of patient, basically perceived exertion was mostly, you can see in all of these patients, they did pretty good on that. And as you can see here, this was the demographics where we had a patient at 21 years, and so you can see a wide range of our patients from young to elderly patients. And disease duration was anywhere from 4 to 10 years. And just looking at the results, participants experienced low levels of soreness after each treatment visit. So that is the numeric pain rating scale that we looked at. You can see that they had low levels, not much different as they were doing, so not significant changes. Participants reported overall tolerance. There was only 1 patient. You can see some wide fluctuations, but most of them did pretty well in there. And as you can see, the patients maintained light to moderate exercise intensity across the various, looking at this across the visits. And also participant increased stimulation settings during, so the participants can actually increase the settings when they are doing this exercise. So they can adjust it according to their activity level. And as you can see, they are actually adjusting it and increasing it as they're doing the exercise. So that, as you can see here, and the results on the right end, you can see the combined single fiber EMG results. This is, I'm not showing you individual one, but the combined, there was some pre-test, post-test changes you can see, although I wouldn't say this is a significant change at this point, but we are still collecting the data. And looking at the efficacy markers, the fatigue severity scale, this is based on individual patient. As you can see, there is improvement in the fatigue severity scale in most of the patients. 6-minute walk test also improved in 3 of the patients. 1 of the patients was stable. And right arm move test, also you can see there's more improvement. And left arm movement test, also you can see some, I mean, again, there's some variability in we are seeing, but this is, as I said, is a small number of patients, and we may need a larger group of patients to look at this more detail. So in conclusion, this preliminary analysis suggests that the whole body electrical muscle stimulation exercise is safe and tolerable for adults with MG, and tolerability, soreness, and these are the safety features that we were looking at. Perceived exertion scores were generally stable across the visits, even with higher intensities. Although they increased the intensities, the perceived exertion was the same. And participant compliance with the intervention was high. 3 out of the 4 participants have expressed interest to continue this if we have a larger study. And data suggests that perceived fatigue is decreased after the exercise with the electrical stimulation. And the data suggests that fatigability is improved after this exercise, and there's improved neuromuscular junction transmission also. There's trend towards that, but we may need more number of patients to, to actually establish that. And again, like we said, we are still collecting the data, and we want to look at more characteristics like age, sex, and along with this, would that change the characteristics of this patient? So that is a study. Thank you for listening. Thank you. And I think Dr. Gao is our next presenter.
Hi, good morning. My name is Shen Gao. I'm from Johnson & Johnson. The first author of Faye is not able to attend the conference, so I'm here to present on behalf of Faye and the study team here. And first, I want to thank the organizer for the opportunity to share this post-hoc analysis in patients treated with nipocalimab. This is our disclosures. And a number of speakers have talked about how important that IgG antibodies are in these autoantibody-mediated diseases, such as MG, especially that a different way of being pathogenic there. Now, modulating the IgG antibodies is now a therapeutic approach, potentially, to treat these different diseases. Nipocalimab is a fully human, high-affinity monoclonal antibody targeting the FcRn receptor. Now, by doing so, it reduces the circulating IgG, including the autoantibodies. The preclinical studies and some clinical studies have indicated that nipocalimab does not impact the IgG synthesis, the antigen recognition, leukocyte proliferations, nor does it impact the level of IgM or IgA. Now, with this mechanism of action, it has the potential to treat a number of antibody-mediated diseases, such as the rare but debilitating diseases like MG, or the maternal-fetal alloimmune diseases, or the rheumatology diseases such as Sjogren's disease. Now, with this potential, we are trying to understand what, in real world, how can nipocalimab impact the other protective immunities that will be very important for our patients. So the goal of this post-hoc analysis is actually the first step of us looking into this. It is to assess the impact of nipocalimab on the pre-existing clinically relevant anti-vaccine antibodies, and also the antibody response to SARS-CoV-2 vaccination and infection. I'll get to that a little bit later. Now, this post-hoc analysis was conducted using samples from the IRIS-RA study. This was a phase 2 proof of mechanism study in rheumatoid arthritic patients treated with nipocalimab. So very, very briefly, this is a placebo-controlled randomized study. Patients were randomized into the 2 arms. They were treated with nipocalimab every 2 weeks, or placebo every 2 weeks for 10 weeks. The clinical efficacy was assessed at week 12, and the safety follow-up continued until week 18. There were numerical improvements in the disease activities at week 12. And actually, the clinical efficacy and safety findings of this study is recently published in a peer-reviewed journal, Rheumatic and Musculoskeletal Disease there. But in the context of this post-hoc analysis, we look at the pharmacodynamic effect here. And as you can see that at this dose, 50 mg per kilo, nipocalimab, it reduces the total IgG by 62% at week 12. And it should be noted that this is the minimal level of reduction of IgG measured at the pre-dose time point. And this 62% reduction is also consistent with what was predicted by PK/PD modeling here, which predicted about 64% IgG lowering, and the same model also predicted about 75% maximal IgG lowering. So this is our sort of reference point in terms of total IgG lowering. Now we first look at the anti-tetanus IgG here. So under current vaccination schedule, almost all adults should have pre-existing anti-tetanus antibodies. In this study, we actually look at the reduction of the anti-tetanus IgG by nipocalimab, and you can see that on the left-hand side, it achieved about 65% reduction from the baseline. That is consistent with what we saw with the total IgG there. Now, if you look at the absolute level of anti-tetanus IgG, so despite the lowering of this, 80% of patients will still maintain a level that is above the protective threshold at week 12, and a majority of patients were protected throughout the entire study period of time. We then also look at the anti-varicella zoster IgG. We saw very similar findings, and there we saw about 61% reduction from the baseline by nipocalimab, and again, a large majority of patients remain protected at week 12 and also throughout the study period of time. The other thing that we looked at was COVID-19. So the study was conducted during the COVID-19 pandemic. There were 4 nipocalimab-treated patients had documented COVID infection. The infection event was noted as the red triangle here on the graphs for the timing there. So among the 4 patients, 3 had mild infections, 1 had moderate infections, all resolved without complications, and none of them required hospitalization. Now, we measured 3 different COVID antibodies, and 2 of them, the anti-spike and anti-S1 RBD, recognizes the spike region of the virus, and they are expected to increase after the vaccination and infection. The third antibody that we measure is the nucleocapsid antibody that we only expect to increase post-infection. Now, if you look at the infection event, the red triangles in these 4 cases, you can clearly see that all 3 antibodies were increased post-infection, suggesting that, that despite the decrease in total IgG level, these patients are able to respond to the live virus infection with the COVID-19. I want to point out that the patient number 1 also has an orange triangle during the study period of time. That orange triangle is a vaccination. So this patient received a 3rd dose of COVID vaccine during the study period. And you can see that they induced an increase in the first 2 the anti-spike antibodies clearly upon the dose of vaccination. There were 2 other patients who received documented COVID vaccination during the study. And patient number 5, it was very similar to patient number 1, had a clear increase in the 2 antibodies that are recognized in the spike region. And for patient number 6, these patients first had developed very high level antibody after the first dose of the vaccination, and therefore did not further increase their level of antibody upon the 2nd dose. So taken together, we know that in clinical practice, there was a variations of response to COVID-19 in both magnitude and the duration, and we saw these variations in the study. But despite these variations, this finding here in these 6 cases consistently show that in patients who are treated with nipocalimab, they can still mount IgG response when encountered with the live COVID-19 virus or with the vaccination. Now, in summary, this post-hoc analysis utilized real-world data from patients who with immunological diseases and treated with nipocalimab. The results suggested that the pre-existing immunity against tetanus and varicella zoster were maintained in a large majority of the patients who were treated with nipocalimab. And when these patients were encountered with COVID-19 virus or the vaccinations, they were able to mount an IgG response despite a reduction in total IgG. Now, we also should know that nipocalimab is not expected to impact the cellular immunity or the IgM or IgAs, so those protective immunity should remain intact with the nipocalimab treatment. The limitation of the study is that this is a post-hoc analysis with a limited sample size, and the finding here needs to be confirmed in the future studies and potentially in additional diseases to further strengthen that. I would like to thank the study participants, investigators of IRIS-RA study. I also wanted to put a plug in here. We also conducted a separate prospective, placebo-controlled vaccine challenge study in healthy participants treated with nipocalimab, and the data are shared here in the session 2 in the poster session. So please stop by if you like. Thank you. And I will introduce our next speaker, Dr. Fei.
Good morning, everyone. Thank you to the organizers for the opportunity to present a small cohort of children that I've followed for the past several years with seronegative myasthenia and autoinflammatory syndromes. These are my disclosures, none related to the topic of the talk. So for the purposes of diagnosing pediatric myasthenia, these are typical criteria for clinical studies. And when patients are seronegative, then we're left to rely on electrodiagnostics and response to acetylcholinesterase inhibitors. And particularly in children where electrodiagnostics can be challenging in the outpatient setting, for many of the patients here, I'm relying especially on the periodistic mean response. So over the past 4 to 5 years, we've collected about 18 seronegative MG patients. All of them have fluctuating weakness, ptosis, and response to periodistic mean. And I've characterized them here by age of onset, weakness profile, antibodies, electrodiagnostic results, genetic testing results, as well as responses to various treatments and associated features. And they fall into several subgroups. There's the ocular group. Not surprisingly, there's a significant number who have pain as a prominent symptom to their syndrome. And then 8 have these autoinflammatory syndromes that I'll talk more about in a moment, with some unusual features that we don't usually see together with myasthenia, including sensorineural hearing loss, gastroparesis, and erythromelalgia. And comparing the seronegative to the seropositive patients, the age of onset, overall age range is generally similar. There's a bit more ophthalmoplegia in the positive antibody cohort and more finger extensor weakness in the seronegative patients and more proximal weakness in the seropositives. Overall, symptom severity is pretty similar, with maybe a trend towards greater severity in the seronegative group. We also use monoclonal antibody therapies more in the seronegative group, probably due to their more severe disease. And then at the bottom, pain was much more common in the seronegative group, and the autoinflammatory syndromes were seen exclusively in the seronegative population. So these autoinflammatory syndromes, so all these patients were previously diagnosed by an immunologist or rheumatologist at my institution with an autoinflammatory syndrome, and these are presumed genetic or confirmed genetic disorders of the innate immune system. They encompass the periodic fever syndromes that you're probably familiar with, such as familial Mediterranean fever and several others. The symptoms can tend to mimic infection and can overlap with autoimmune diseases being more the disorders of adaptive immunity. And so you can see fevers, migrating joint pains, muscle pains, rashes, and occasionally more unusual findings like aseptic meningitis, sensorineural hearing loss, and eye disease. Inflammatory markers are frequently elevated during episodes or even interictally. And then treatments tend to be either on the broader spectrum, steroids, colchicine, or targeted treatments for the IL-1 and TNF-alpha pathways. So as I mentioned a couple slides ago, we diagnosed 8 patients with seronegative myasthenia with autoinflammatory syndromes, and I've detailed 7 of the cases here. Interestingly, there are 3 sets of siblings, 2 sisters, a brother and a sister, and twin brothers. The 2 sisters both have relapsing polychondritis, which is a subtype of autoinflammatory syndrome. One of the sisters, the older one, has been probably my most challenging patient over the last few years. She's required frequent plasma exchange, and we've tried multiple monoclonal antibodies. And her sister is less severely affected and quite responsive to rituximab. We have a brother and sister who have cryopyrin-associated periodic syndrome, which is caused by a mutation in NLRP3 gene. They both have gastroparesis. The brother has been quite refractory, has had a partial response to efgartigimod, but has required frequent IVIG and plasma exchange as well, has been intubated 3 times. And the sister is much more mildly effective and rituximab responsive. We have a 9-year-old boy with various skin rash symptoms, migraine, erythromelalgia, and gastroparesis. His repetitive nerve stimulation was completely normal, albeit just 3 nerves were tolerated when we did the study. And the patients above, the sisters had clearly abnormal repetitive nerve stimulation, and the brother and sister were kind of borderline. And so the 9-year-old boy was very rituximab responsive, although we had to switch to obinutuzumab later. And then the twin brothers both have sensory no hearing loss. Interestingly, one of them had lost hearing completely before I met them, but the other's hearing loss physiology correlates quite nicely with IVIG dosing. So you can see his the measures of hearing actually improving with each IVIG dose. They both have fatigable hand weakness. One of them has erythromelalgia, which has also been responsive to IVIG, and one of them is eculizumab responsive. So this is just to summarize the monoclonal antibody treatments that have been effective or partially effective in this cohort, including rituximab in 5, eculizumab in 2, efgartigimod partially in 1, obinutuzumab in 2, and daratumumab, plasma cell inhibitor in 1. So revisiting the diagnostic criteria in the seronegative group, where we're reliant on abnormal electrodiagnostics in response to pyridostigmine, and especially when the electrodiagnostics are a bit equivocal, I've been able to feel more confident moving forward with treatment of these patients through confirming that there's no congenital myasthenic syndrome through genetic testing, making sure they have weakness at baseline and throughout treatment in muscles characteristically affected in MG, and then I take a gradual course moving from steroids to IVIG and then on to monoclonal antibodies. And so this has given me more confidence in the diagnosis, and I suspect that overall these children with autoinflammatory syndromes are underdiagnosed because their fatigue and weakness are often blamed on other autoinflammatory symptoms. But overall, they seem to have very similar clinical features to the seropositive patients, and they can have quite severe disease. A couple of them have required prolonged respiratory management, but they do seem to respond well to both B-cell and complement-targeted treatments. And so we have ongoing work now trying to better characterize the common pathways for autoinflammatory syndromes and MG. We're doing some antibody discovery through phage display with some collaborators on campus, and working on getting sedated single-fiber EMGs for some of the more patients with more equivocal electrodiagnostics. And hopefully this will allow us to treat this population more effectively. So thank you for your attention. And Dr. Novak will give the final talk.
So thank you again. So what I'll be doing is presenting the exploring outcomes and characteristics of myasthenia gravis 2, which is Explore MG2, design, rationale, and baseline results. So I'm presenting on behalf of my colleagues in MGNet today. So these are my relevant disclosures. So the background here, Explore MG2 was initiated as one of the core programs of MGNet. MGNet is funded by the National Institutes of Health through their Rare Disease Clinical Research Network. The purpose here was to utilize 2 previously established and ongoing registries with biorepositories, which were Nimble at Duke and Explore MG at Yale. Explore MG2 utilizes NIH common data elements for the study of myasthenia gravis. Our main objective was to better understand the disease characteristics and to link these to mechanistic markers of disease activity. Our methods were to have a detailed prospective characterization of the natural history of myasthenia gravis, including deep clinical phenotyping of a largely neglected MG subgroup, such as seronegative myasthenia gravis. And our ultimate goal here was to couple the phenotype to biospecimen collection so that we could study this disease in the future, and actually also try to identify treatment-responsive biomarkers and other projects along the way. So initially, patients were enrolled with baseline visits, which captures the basic demographics, disease history, diagnostic data, comorbidities, outcome measures, and biomarker samples. The purpose here was to study patients for a minimum duration or a total duration of 2 years with samples collected at a minimum of every 6 months, with clinical data collected at a minimum of every 6 months. Sites were also encouraged to enter data in between visits, so we had a very robust data set for study in future. At follow-up visits, details about their myasthenia gravis were entered, including medical history, changes in MG medications, outcome measures, biosamples, etc. So our initial target enrollment was 400 patients, and our goal was to enroll patients within 2 years of their disease diagnosis. So it's difficult to enroll treatment-naive, newly diagnosed patients, so we had a pre-specified disease duration cutoff for enrollment, with the exception of more largely rare conditions such as MuSK, which we didn't have an age restriction or a duration of time restriction for enrollment. This details sort of our natural history data collection, which was pretty busy, but it includes MGFA clinical classification, ADL scores, QOL scores, EMG results, antibody status, etc. And you can see at our follow-up intervals, a number of different outcome measures were collected. So enrollment is now completed as of the end of May. We enrolled just over 300 study participants. So we say, well, I presented 400, why did we do 300? So in the setting of the COVID-19 pandemic, we had a lot of research pauses. So we needed to recalibrate our total study enrollment goals to account for that. And we utilized some of those resources to fund a telemedicine study. So our overall enrollment rate was 7.4 participants per month. We utilized a total of 10 sites. We had 6 core sites with the addition of 4 ad hoc sites during the course of the clinical trial. So it represents a multi-center study of sites in the United States. So follow-up is still ongoing despite the study being now complete to enrollment. 19% of our study participants have completed the 2-year follow-up period. This details the number of biosamples that we've collected over the course of the enrollment period. And so you can see how the numbers keep sort of dwindling down. It's mainly because the patients are still in study and we're still collecting those data points along the way. So I'll just leave that there. Let me just jump into some of the characteristics. So 43% of our population was female. Age at enrollment was 59. Average age at diagnosis was 56. Our mean MG-ADL score is 3.8, and our mean MG composite score was 6.2. And these are all snapshots at time of enrollment or baseline visit. Our antibody subtypes, very similar to what's reported in the literature in terms of the breakdown. We have about 72 or so percent with acetylcholine receptor autoantibody positive subset, 14% roughly with MuSK, about 13% seronegative, and 0.3% of our population is LRP4 positive. In terms of additional demographics, race, and ethnicity, the cohort is represented by the majority white with 78.5%, followed by Blacks, African Americans at just over 10%, and then Asians at 3.9%. Ethnicity, again, favored non-Hispanics at 89.5%. So if you look at the US census data, it's not totally representative of what the US Census race and ethnicity breakdowns are. It's underrepresentation of what we typically see, but there is representation in all of the various subcategories. So it is quite diverse as a cohort. So I'll just conclude here. So the Explore MG2 is the largest prospective longitudinal natural history study with a complementary biorepository in myasthenia gravis to date. The goal was that this would improve clinical trial readiness and to facilitate development of treatment-responsive biomarkers. So early in the morning, there was a lot of discussion on biomarkers, things that might predict response to treatment. How do we better subtype beyond, say, antibody status and other things? So we're hoping that this data set, once it's completed in full, will be used as a tool to understand the disease and the natural history of autoimmune myasthenia gravis. It's modern, multi-centered. The goal here is to understand the mechanisms of disease and to also provide an ability for biomarker discovery, biomarker validation, and future translation to precision health. So please visit the following website for more information about MGNet as well as our data and biosample sharing policy. So we encourage you all to reach out about the potential for collaboration and the use of the data. So the intent of the collection of the data set and the biorepository is that we could share this and collaborate with the community for future investigation. So thank you for your attention. And it's either going to be Sam? Okay. So I'm going to hand it over to Sam Masterson, MGFA CEO. So thanks very much.
All right, I'm going to wrap up quickly. I know you all have other sessions to get to, but I have to say I always feel like getting up here and saying we made it, because it's such a fast 4 hours, and I know you guys, it's like most of them are 7-minute bite-sized presentations, and I just really appreciate you bearing with us and staying until the end, so thank you very much. I hope you enjoyed it. I hope you learned a lot. I hope it sparked a lot of curiosity and made a lot of new connections as well. That's an important part of it. So I do want to thank our committee members again. We have a couple sitting right here. Dr. Castro had to take off, but could you guys just stand again real quickly? I'm sorry, I just really want to acknowledge you. Thank you. I want to thank all of our speakers. Each year I thank everyone for their attendance and their support, but I think it's really important to acknowledge that we thank the speakers, but we also want to congratulate you for your invitation to speak and to present. And let me just give you a snapshot of how much this scientific session program has grown. In 2021, we had 27 abstract submissions. In 2022, we had 39. In 2023, we had 76. And this year, we had 122 abstract submissions. So congratulations to the speakers for being selected. Yeah. Again, thank you to our generous sponsors. We appreciate the support so much. And I wish you all a wonderful rest of your AANEM meeting and week and travels home. So thank you.